and specific aspects of something coming up here. Specific aspects of emotional processing, and damage to one hemisphere can affect motor function on the opposite side of the body, and this means that damage to the left hemisphere can affect motor function on the right-hand side of the body and vice versa. The primary objective of neurophysics therapy when treating stroke and ABI patients is to lateralize the hemispheres and enriching the environment along with specialized methods to assist the individual to compensate for their lesion and to regain a centered experience of their world. So what is neurophysics therapy? Well, neurophysics per se is a branch of medical physics and science dealing with the development and use of physical techniques to gain information about the nervous system, usually at a molecular scale. So functional MRIs and PET scans are usually used to do this and to study perception, action, and cognition. And action and cognition and neural networks and complexity science are just some of the tools utilized to make sense of the data and develop theory. Neurophysics therapy per se was founded more than 35 years ago, and I'm very proud to say that it has earned its own footprint within this branch of science. And neurophysics therapy is the study of the functionality and behavior of the human nervous system and how it perceives and responds, interact with the environment, and the development of specialized techniques to optimize the system's functional and behavioral potential to accomplish specific outcomes. So being relatively successful over the years, and there's been a lot of media, international and national media attention attributed to many of the patients that come from overseas and nationally who've achieved incredible outcomes, and so the likes of 60 Minutes and BBC. As a matter of fact, as I'm speaking right now, there is a national television story about one of the other patients that we've been working with, a young 15-year-old boy who was born with a tumor on his spine, and he's been able to move his legs quite significantly voluntarily. So the story is airing as I'm talking now. So this is all culminating in the Academy Award nominee Stephen Pazwalski's multi-international award-winning documentary Calibrate. So it's a beautiful film about people who have come from all over the world for the therapy, telling their stories and what they've accomplished and so forth. So if you ever get the opportunity to see it, like I said, it is a very beautiful film. So here's some past and present stroke testimonials, basically from stroke patients that we've studied. They're credible sources here. Pictures has 1,000 words. So within these videos and testimonial videos, we see a lot of the descriptions of the function, and in particular, pointing out the fact that these incredible results can occur within 4 days of neurophysics therapy. So basically unprecedented. So we'll go to that, and then we'll carry on with the presentation.
So I had a stroke a little over a year ago, and I work for Gatorade, and I had worked with a colleague, his name is David Knight, who used to run Gatorade in Australia, and he was friends with John McLean, who had been helped by Ken and Neuroscience. So they suggested that I contact Ken to see if they could help me with my walking and give me back functionality of my left arm. So I had a Skype consult with Ken about 2 months ago and made arrangements to come in to see if he could help me regain more functional use of my arm and my left leg. So I was originally completely paralyzed on the left side, and I've been having therapy for the last year, regained some functionality, but still walk with great difficulty with a cane and not able to move my left arm at all. And a strange side effect is that my left fingers are always clenched very tight. And that's one of the things we've been able to see in the week that I've been here, that I'm able to walk a little bit without my cane. And I'm seeing the fingers start to unclench in the time that I've spent with neurophysics.
What are some of the things that you've noticed that may have improved since you've been here?
Because I've had a pain in my left shoulder since the stroke, and I've seen that lessen some. And the ability to walk without the cane in a more upright fashion is something that's very different. And these changes I didn't see from one session to the next, but as I look back on the week and look at my movement now, it's so much better now. It's day and night in 4 days to go from not being able to walk anywhere without my cane to now being able to take quite a few steps without the cane. and being able to move my arm more than I have in the past year. So that's phenomenal progress from where I started.
What did you know about neurophysics beforehand? You said you heard from John McLean through a co-worker, but after researching it a little bit and speaking with Ken, what did you think of the therapy and how it would work?
Candidly, I'm still very confused about the therapy, and I wasn't sure how it was going to work. And initially, as I watched videos and read about the therapy, I expected a miracle, but then I read John's book and realized that this is something that's going to take a little bit of time and a little bit of work. So I came in prepared to put the work in. And I was a little confused as to how they were going to induce the tremors. My first session here, I didn't get the tremors, which I was very disappointed because I didn't get them, at least until the third session when I started to experience the tremors. So I was a little concerned, but by the fourth session, I was walking without the cane. So that's certainly something that after a year, it's very surprising that I would see that difference in 4 days.
How would you explain the therapy?
It's, what Ken tries to do is to get you to relax as he's getting you to lift some very light weights and think about your body being cut in half and I'm impacted on my left side, so my left side needing to learn from my right side and relaxing and stopping my right side from being so dominant. So that need to relax and let the healing happen is something that's very different and very difficult to do. I've signed up for a half marathon in November. That might be a little optimistic, but I think it's good to have these stretch goals that subconsciously, that tells my brain, I think, that we've got work to do and that the right side has got to stop compensating for the left side and let the left do what it needs to do. So that's what I'm optimistic about in the next few weeks.
For you to come out here to do this sort of therapy, you must really think that it can be very beneficial.
Well, it's hard, even though you don't understand how it works. I didn't understand how it works. It's really hard to argue against the results. And as a scientist, I think there are a lot of discoveries which, when they happen, there's often the mechanism is not clearly understood. So for me, as long as the results were there, then that gave me the confidence to come in and put myself through the process and trust that whatever the mechanism is, whether it's understood or not, that if I would participate in the process as designed, that I should expect the same kinds of outcomes. And that's what's happened. And I think what Ken's doing now is he saw the outcome before he understood the mechanism. And now he's understood the mechanism and is getting the rest of the world to think differently, which is typical for true breakthrough innovation, that it happens and only a few people get it, and they have to take the time to bring the masses along. And I think that's what Ken's trying to do right now. We have so many redundancies in our bodies that our body tends to find ways to, survival, you've got to have more than one pathway. So it wouldn't make sense that as a species that we would only have one way of doing things. And I've read a little bit about neuroplasticity, so I have real belief that this is something that happens. And for me, maybe my learning mode is such that understanding what Ken's trying to do makes it easier for me to accept the learning, because most of the therapy that I've gone through has not really done much to explain the underlying, processes with the brain and what's going on. It's just more repetition of particular activities, and that for me has not been as effective as trying to engage the brain in sort of rebuilding or repathwaying itself.
What would you say to somebody who is considering coming and trying neurophysics?
I would say you have nothing to lose except whatever problem you're dealing with. So just go ahead and do it, because chances are, if you're considering this, then whatever your work you're doing is not working as effectively. So part of insanity is doing the same thing over and over and expecting a different outcome. So I would say if you have something you're trying to resolve, this is as good a place as any to come in and experience and commit to solving the problems at hand. You have absolutely nothing to lose and everything to gain. I'm going to be dancing in a few weeks, I hope.
2007, I suffered a grade 9 brain hemorrhage due to brain aneurysms and subsequently 4 strokes. I've been paralysed since 2007. No one in the medical fraternity has been able to make any improvements with me, but recently I discovered Ken Ware's Neurophysics, and they've just helped me immeasurably. I couldn't walk properly, I couldn't use my arm or limbs properly. Now they're all working again like I'm normal. It's really built up my confidence, and I just feel so much. I feel like a normal, different person again. Yeah, I couldn't even open my hand or grip anything. Now I can do everything with it, and I've got all my functions back. I couldn't walk properly, and now I walk like I'm floating. It's just sensational. I think they're more in touch with connecting the mind and the body together, and that has really helped me. I've tried acupuncture, I've tried many different therapists, from physiotherapists to chiropractors and different neurosurgeons. Most of them sent me away and said, "Oh, you're too far gone," and that's it. Ken and Nikki have been the only ones who have actually made any progress with me and helped me. This is the first improvement I've ever had since 2007. The exercises I've been taught to do have just changed my body for the better. And now I can move forward in my life and go forth with freedom and do all the things I wanted to do. And perhaps I might even go back and make some more wine, because I couldn't previously. It's stopping me from being able to do my regular work, which was winemaking. So now that I have my functions back, I can continue on in my loved career, so to speak. I never expected to come out of the therapy as functional as I have. I'm totally overwhelmed. It's the best thing that's ever happened in my life. It's improved my whole well-being. I've got feeling back in my body. I can feel everything in my body. I can feel my hand again. I couldn't feel my hand since 2007. I couldn't feel my leg. Now I can feel my leg again. It's absolutely amazing. I'm astounded. Through the therapy, I've actually gained a hell of a lot better in my health. I'm a lot fitter, I'm a lot healthier, and I just feel more whole as a person. I'm definitely more connected to my body and in touch with my mind and my feelings. But after the therapy with neurophysics, I have no pain whatsoever at all. And I'm not on any pain medication. I feel free and to live my life. In 2007, I suffered a grade 9 brain hemorrhage.
So neurophysics therapy and stroke and acquired brain injury. So neurophysics therapy is that form of therapy that by design can begin to quantifiably restore motor functions in stroke patients and patients with acquired brain injuries in very small time scales. So we actually have that 4-day measuring stick where the expectation is that significant changes are going to occur in that 4-day period. But it may make note of the fact that patients are required to be cognitively aware. So they need to be able to apply themselves. They need to be able to perceive their environment, act upon the environment, and then be able to cognitively manipulate the outcome. So neurophysics therapy uses select pieces of unilateral exercise machines to promote movement symmetry and sensory feedback, which has proven to be highly successful for rapidly promoting motor functions in patients with neurological conditions. And NPT works by assisting the patient to recognize feedback feed-forward sensations on the affected side of their body and focus on lateralizing the experience towards sensations of sameness, mirroring the unaffected side of the body. A vital component of NPT is exploiting the central pattern generators to promote synchronized bipedal limb movements and promoting up organization of communication within relative assemblies of synapse. However, it is very much the policy to treat the person, not the disorder, through the lens of neurophysics therapy, and that's across all types of pathologies that we deal with. So individual differences among human beings disallows for generic engineering of therapies to treat stroke and brain injured patients. And this is one of the stark differences between NPT and other modalities, and pivotal to the high success rate of NPT. And as I say to my students, people don't care about what you know until they know that you care. So when treating the person, not just the disorder. Several aspects are emphasized. So a personalized treatment plan. So each person needs and circumstances are different, and their treatment should be tailored accordingly. So an NPT practitioner will assess the individual's physical, emotional, and cognitive state to develop a personalized plan that addresses these specific needs. Functional exercise, and NPT encourages the use of controlled functional exercise that stimulate the nervous system to create more beneficial neural pathways. And these exercises can help individuals build strength, flexibility, and resilience, while also promoting mental and emotional well-being. And of course, a lot of people say they have used very similar language. The only difference is with NPT, the expectation is that results come along quite rapidly as opposed to months or even years. So integration of the psychophysical brain-body, so not mind-body. NPT recognizes the interconnectedness of the psychophysical brain-body and works to address both aspects in treatment. And by fostering a deeper understanding of the psychophysical brain-body connection, individuals can learn to better manage stress, emotions, and physical symptoms. Empowerment and self-regulation. NPT aims to empower individuals by teaching them self-regulation techniques, and by learning to listen to their body signals and adjust their movements accordingly, individuals can take greater control over their health and well-being. The continuous adaptation. NPT acknowledges that change is a constant in life, that an individual's needs may evolve over time. Consequently, treatment plans should be continuously adjusted to ensure ongoing progress and improvement. And by focusing on the person as a whole rather than just the disorder, NPT aims to facilitate lasting change and improve well-being. This approach empowers individuals to take control of their own healing journey and fosters resilience, adaptability, and self-awareness. Noisy inputs infers noisy outputs, and it is more often the case that much effort is required by the patient to detrain existing patterns of functionality and asymmetric dominances that have arrived post-trauma. And reserved in the neurologically compromised patient, that increases anxiety, amplifies symptoms effect. This creates a positive feedback loop. As the symptoms increase in value, the anxiety increases in value, that increases the symptoms and so forth. So neurophysics therapy involves a super slow self-controlled hormesis-like perturbation to the patient's psychophysical system via the use of resistance exercise equipment within a safe, supported environment. This exposes the patient's underlying fears and anxieties and identifies the patient's rogue sensory perceptual default responses to environmental stimuli. The patient is perceiving non-noxious environmental stimuli that they have control over the input of as noxious. And these are the sets of initial conditions that give rise to rogue psychophysical systems behaviors. Once the patient can modulate their limbic system and all its connections to detrain their rogue perceptual errors, a simultaneous relaxation of symptoms occurs with a normalization of cognitive processing. The purposely prescribed perturbation is repeated, and the patient is encouraged not to inhibit the emergence of any neural tremor, and instead to allow these underlying kinetics to increase in value chaotically. This can be initially confrontational to the willing and trusting patient. However, the patient participates and cognitively learns to let go and enable these chaotic dynamics to evolve. We observe a desirable change in the structure of these time-dependent dynamics, coinciding with an increase in velocity of movements across all moving parts of their system, as their system disposes of its overorderliness and self-organizes to higher states of complexity. Disease and disorders is viewed as a loss of the system's complexity. So neurophysics has proven to be highly effective for patients who are prepared to render their own healing through this evolving process. So here we are just simply expressing how the central nervous system can, when given the right sets of initial conditions and through lateralization of the hemisphere and the enriching of the environment, compensate for extreme lesions to it. So here we have Kylie. She was an elite athlete. They thought she'd torn her hamstring and they were treating it as such. They waited until this tumor had a 2-litre blood supply before they viewed it as suspicious. This came about the removal of her whole sciatic nerve, her hamstring, 3/4 of the adductor and 3/4 of the adductor process. So it was a given, as far as the medical experts were concerned, that Kylie would never be able to voluntarily pull her leg back, use a hamstring basis, use that leg to help her sit down or stand up again, bend her knee effectively or walk properly. And as well, she was going to experience long-term a lack of sensation below the knee. However, in less than 2 months in neurophysics therapy, Kylie was able to demonstrate sound bilateral stability when doing difficult exercise involving both limbs. As demonstrated, her gait dramatically improved and significantly normalized, and she now has constant sensation in her lower left leg beneath the knee. She was soon able to jog on uneven ground cross-country for up to 5 kilometres. So here we can watch Kylie going through some of these movements, very much nice and relaxed and composed, thinking about being nice and centered with the movement, and just monitoring the sensations left to right as she's going through. So we often need to close the eyes because the visual system has a bias at times and can very much persuade the system back to some pre-state. So we see this exercise of bipedal movement as such. You see great lateral stability in the left leg, despite the fact that 3/4 of that adductor process has been missing. So the role proprioception plays in the acuity of our perception of the environment and relationship with it. So one of the first things we do when we're treating patients is to check their proprioception. So proprioception is just nested within our perceptual landscape as to what we perceive to be a reality. So if we see errors in proprioception, it's given that there's errors at all other levels of percept for the person, and not that they're purposely going out and making false perceptions. They see things for as they are, hear things for as they are. Because as that information gets downloaded into their system, because of stresses and other types of inhibition, that's when the sensory perceptual errors will occur. And they all need to be corrected in every single person before we progress with the therapy. So that's part of the lateralization of the hemisphere. So if we have a person who's redundant on the left-hand side of their body, has been dramatically affected, we'll still use whatever we can with the right arm, touching the knee with their eyes closed, very slowly moving to the point of the nose. And so just to sort of prune that in, we'll often see errors. But as that person learns to prune those things, we see that neurons in the brain are now firing differently in a much more responsible way and coordinated way. So in summary, proprioception plays a vital role in shaping our perception of the environment and our relationship with it. It allows for us to accurately perceive our surroundings, that respond effectively to our environment, and disruptions to the system can lead to inaccurate perceptions and impaired relationship with the environment. So central pattern generators, they're neural circuits located in the spinal cord and brainstem that control rhythmic motor patterns such as walking and swimming. And central pattern generators produce synchronized bipedal movements such as walking. By exploiting central pattern generators, neurophysics therapy can promote synchronous limb movements and improve motor function in patients with neurological conditions. So CPGs can receive input from higher centers of the brain, including the frontal lobes, premotor and motor cortex, cerebellum, and therefore they can be influenced by supraspinal structures, and that's very much where we come in and play a major role in the exploitation of that at that level. So being in the zone, exploiting the concept of decentralization, which is what we certainly subscribe to. However, a lot of the work on central pattern generators, there's an enormous amount of study going into central pattern generators, and a lot of times there's a lot of conflicting information about it. However, the decentralized approach is very much an approach that is used for the development of robots that can navigate their environment and compensate for different things as they come along. So they're saying this reduces the time delays in motor control loops where the rhythmic motion is controlled by short feedback loops, etc. And again, we're looking at a very mechanistic way of looking at that. However, a robot will never be able to surf pipeline in Hawaii, skate like a world champion, do extreme parkour, or perform elaborate slackline tricks like a human being can. And the variances in mental states during performance of a human being affect outcomes. So we see these people here doing some amazing things where they're just in the zone, they're just navigating their environment. Tennis players, the whole lot with that extreme decisions need to be made very rapidly. And a lot of times when we see that the information arrives of a person doing parkour, running through very foreign, strange landscapes, responding to that environment at timescales, very appropriate timescales, it does challenge a lot of the models. And perhaps the conscious electromagnetic information field theory, which involves olfactory communication, may assist in a great understanding of how a person can instantly respond to foreign and complex environment stimuli in such a seamless manner. So neurophysics therapy process exploits central pattern generators. And again, it's about being in the zone. Executively, remaining calm and relaxed while thinking about flow and distribution of the sensory experience across all scales of the system during the execution of the prescribed exercise movements allows for the progressive removal of the system's restraints, enabling the system to self-organize to higher states of complexity. So the machines are actually unilateral. Some of the machines are independent on cables. And it's the idea of the person being able to, and we help them a lot to get that feedback from initially, and eventually they can do that by themselves in very small timescales. So exploits pattern generators by restore motor function by using simultaneous movement in both limbs to initiate mild, a super slow stimulus to the psychophysical system, and transitioning to bipedal movements as mirroring of these connections strengthens. So the use of these particular machines, by using these machines, patients can begin to regain awareness of a centered bilateral equal experience as the limbs very slowly move in a synchronized manner at each point in space-time, which modulates asymmetric or loss function. Early intervention is very important if possible. So early intervention can also prevent compensatory movements from becoming ingrained and impeding the restoration of motor function. In most cases, NPT process begins by detraining compensatory movements and overworldliness, excessive tone in both the unaffected limbs and the affected limbs, and in sympathetically overtoned muscle groups such as the trapeze to enable flow of information and an even distribution of sensory information to occur across all scales of the system. This needs to be done in a slow and meaningful manner with lots of coaching of the patient to cognitively apply themselves with these executive focuses. So there's ongoing research and development, the role of lateralization of the hemispheres, proprioception, and even posture play in the restoration of lost function and the maintenance of a healthy psychophysical system. So we use a variety of technologies, EMG, ECG, and EEG, along with thermal imaging and zebra stance and gait technology. This enables us to cross-check the reliability of any device compared to others in order to validate the effectiveness of NPT in the treatment of all complex psychophysical diseases and disorders and complex lesions of the nervous system. So here we have a neurologically compromised patient seated at rest day 1 pre-neurophysics therapy, some data, channels 1 and 2 EMG, left and right trapeze, channel 3 and 4 EMG, left and right abdominals adjacent to navel, channel 5 and 6 EMG, left and right adductors, channel 7 the ECG, and channel 8 the EEG premotor and motor cortex, the left hemisphere for this particular person. So these data display the random systems-wide overworldliness and highly synchronized events occurring in these patients' psychophysically sensory motor system with obvious differences between the left and right side of the body. The emergence of these symptoms was sensitive to the system sets of initial conditions, which initially involved perception of the environment. Here's some compelling comparative data. So on the top left-hand side there, that is the data we were just viewing. On the right-hand side there is a fit, healthy control at rest. So we can see that there's a nice even distribution taking place across all channels there at rest pre-day 1. And down the bottom left-hand corner, we see the neurologically compromised person at rest day 1 post-neurophysics therapy. So we see things are starting to come back online and balance out a lot more. And on the right-hand side there, we see the neurologically compromised patient at rest day 4 post-neurophysics therapy. So we can see there's been quite a dramatic normalization within these data. So thank you very much for your attention, everybody. And again, I was very privileged to be able to do this presentation again. And I wish everybody attending the meeting and doing presentations all the very best with their presentation.
Yeah. Thank you, Dr. Ken. Is there any questions from the audience? Yes. If there are no questions, then we can move ahead and for the next presentation. Yeah. The next presentation is Dr. Peter Nara, Keystone Bio Incorporation, United States. Dr. Peter, can you share your screen, please?
Sure. Can you see it? Can you see it okay?
No, no, doctor. Can you share screen from Zoom?
Yeah, this is the one I have.
I'm that's fine. Like I think you're opening the presentation slides in your system. Instead of that, can you open in share screen in the Zoom meeting? You're unable to see your screen.
Oh.
Can you just open the Zoom meeting?
Yeah, I did.
There you can, yeah, in the bottom you can share screen and particular file because that file is not viewable for us. We cannot able to see.
Yeah, I'm
Yeah,
sharing it.
that's fine now.
You can see it?
Yes, but we are able to see Neurophone. Yeah. So open that. There is your presentation. Yeah. Yes.
Can you see?
Can you click on it?
Can you see
Yeah,
that?
yeah. Yeah. Can you press
Okay.
F5? F5, yeah. Thank you. You can start your presentation.
Okay. Okay, thank you.
Yeah.
Yes, good morning, everyone. It's a pleasure to join everyone, at least virtually. Hope things are wonderful in London today, and I'm excited to talk to you. I am the Chief Scientific Officer at Keystone Bio, that's here in St. Louis, Missouri, in the United States, and be sharing with you what really is a compilation of many years of studies by many individuals over much time on trying to identify the sources of major neurosystemic inflammation in the body. And today I'll be discussing with you the really great advancements that have been made in identifying a singular bacterial cause and portions of the bacteria that in fact themselves are produced that lead to these diseases. So the talk summary is going to be covering this bacteria called Porphyromonas gingivalis. We have termed it a biological extremophile, and it appears now to be the major etiology for neuroinflammation, systemic inflammation leading to dementias and Alzheimer's disease, as well as Parkinson's disease. So most neuroinflammatory diseases that otherwise don't have an etiology now seem to be driven by this organism and its biological byproducts. Koch's postulates have been completely fulfilled for this, indicating the causative agent at this point. I'd like to review the preclinical scientific evidence, review some of the clinical scientific data, and then update everyone on the clinical development of a specific biotherapeutic and companion diagnostic for it. So the literature over the last 20 years or so now is pushing almost 10,000 publications on this organism. It's garnered quite a bit of attention, as you can see here by the cover of the New Scientist a couple of years ago. It is now in medical diseases, has over 1,400 citations. In the PG-driven inflammasome and inflammation space, there are 1,800 citations, 177 published just last year. On PG and dementia and Alzheimer's now, we're at 142 citations over the last 2 years. And in oral esophageal cancer, 328 citations now. Someone might look at this list and say that's an extraordinarily broad medical symptoms list and conditions list, and we agree, but there is a central thesis to all of this. So the oral bacteria, as you can see here on the left, it sits in the gum line, below the gum line, and it's deep within an anaerobic tissue chambers in the mouth, infecting most, if not many, teeth over time. It's present in about 40 to 80% of adults, depending on their age by decade. Locally, there is a fair amount of A beta amyloid that are made in the gums, on the gums, and in the liver of individuals infected with this oral PG infection. And it actively creates a biofilm that is polymicrobial and is essentially impossible to remove. The problem is that it doesn't just cause local damage here in terms of its oral dysbiosis. It is a champion bacteria at releasing outer membrane vesicles that are laden with bacterial toxins, which are then released in these structures called outer membrane vesicles, which have ultimately become additionally immune complexed. These course throughout the systemic circulation and end up in various end organs, particularly getting off in the blood-brain barrier, the cerebral meningeal vessels, the liver, the heart, large vessels, the pancreas, and so on and so forth. And so as you can see here, there's a fairly broad distribution starting from the mouth where these PG toxic protein outer membrane vesicles end up. We're going to focus today particularly on those occurring, driving neuroinflammation, neurodegeneration. Just to know that the cardiovascular, atherosclerosis, systemic sclerosis effects are well recognized, is to show you here the systemic wide inflammation that's specifically driven by PG and has been measured as increased in individuals with biomarkers panel testing. And you can see that the major large biomarkers for inflammation are activated. Reviewing the neurodegenerative disorders, as I mentioned, Alzheimer's disease, vascular-associated dementia, Parkinson's, Downs, stroke-related dementia, microvascular defects, peripheral amyloid beta, age-related macular degeneration, and neuroinflammation related through TNF and IL-1 and NLRP3 inflammasomes. So in 21, we released a major paper trying to pull this all together from our own work and that which existed in the Alzheimer's disease literature. And I'll refer you to that publication. But it was, we were able to bring together what at this point was an unexplained relationship for the neuropathogenesis for the cholinergic hypothesis, the iron dyshomeostasis model, and salivary lactoferrin in Alzheimer's disease. Outer membrane vesicles were able to explain the results and the outcomes in Alzheimer's disease in these cases. As you can see here, we have an increasing prevalence of infection with Porphyromonas gingivalis as you get older. There's community-acquired infection starting in the 30s to 40%, in the early 30s to 50, 60, 70%, and higher over age 60. What's important to note is that we now understand that PG exists in virulent, hypervirulent, low virulent, name virulent subtypes and genotypes. And before this, there was less appreciation for the complexity of the data that was gathered, leading to some studies not clearly proving associations and drivers of disease, which once we sort out those that are virulent, more virulent with disease, of course, then we have high specificity for causation. Here's a little bit closer view of these outer membrane vesicles. They're elegantly structured lipid bilayers that come off the bacterial surface. And these are literally have their own proteome and they have a small genome. Right now, they're based on mass spec analysis, at least 150 or 151 proteins that are packed within the vesicle lumens, many of which are lipoproteins and proteins of various types. These are essentially communication vesicles for the bacteria distant site and modulate all kinds of things in the environment, both locally and at a distance. The studies that have been done recently, published here this past year on the animal model, looking at PG outer membrane vesicles, once they're infused, they rapidly find their way to the brain and through the blood-brain barrier, through the endothelial cells. And most importantly, in a short amount of time, they decrease the tight junction related gene, protein, and gene expressions, as you can see here, as compared to control OMVs. In addition, in these studies, these PG OMVs as compared to controls increased tau phosphorylation in the hippocampus region. Again, a classic hallmark of Alzheimer's disease. Here you can see control as opposed to OMVs. Additionally, OMVs induce neuroinflammation, as mentioned here, studying IL-1 beta, GFAP, and IL-1 beta, again, compared to controls. Rather striking induction of neuroinflammation within a relatively short period of time. These don't require the blood-brain barrier in this case to have been broken down, but these vesicles can transmigrate and get into the parenchyma, causing additional damage to neurons, astrocytes, and microglia. More importantly, also these PGOMVs trigger the NLRP3 inflammasome in the hippocampus of mice as well, as you can see here, as compared again to control, capase-1, ASC, etc., both intracellular and extracellular. So if we look at the schematic version of this, we can see PGOMVs, they're rather taken up very quickly within minutes. through an intracellular inflammasome activation pathway, as laid out here within microglial cells, endothelial cells, and ultimately in then activating IL-1 beta processes in the brain, which then have extracellular effects, of course, on neurons, as you see, tau phosphorylation, neuronal degeneration, and memory impairment. This is an example of the actual toxic proteins that are in those outer membrane vesicles that have been observed by histohistochemistry using one of our monoclonal antibodies that are used as the biotherapeutic to demonstrate the relative intensity and fluid distribution of these particular toxins here within the dentate gyrus of Alzheimer's disease patient, the lower right being the negative control. Here you find hippocampal neurons and microglia. Again, low mag showing the rather diffuse distribution. And then the positive punch biopsy from the gum showing the florid amount of the same protein made locally within the oral cavity, and then, of course, ending up throughout the specific neuroanatomic regions of the brain. To confirm that these were, in fact, the real proteins, there were pull-down experiments done with mass spec to identify fragments of the protein that were in those particular immunohistochemical lesions. In addition, there was a lot of discussion and studies done by other groups that suggested that this may have been a translocation of an active infection from the mouth to the brain, with there being an active brain infection driving most of the neuroinflammation for Alzheimer's disease. We tested that in a total of 61 total brain specimens, 31 of which were negative, 7 of which had very few copies. And so our overall summary for this was, in fact, that there are not enough samples positive for bacterial DNA to suggest that there's any active bacterial infection within the brain, but in fact that many of these that are positive due to the outer membrane vesicles having transmigrated to the brain. And as I showed you before, this shows additional immunohistochemistry. Here we see the choroid plexus lit up as well. What was interesting is, independent of our work, it was a recent summary and release of data from a clinical study that was done by another company studying a gingipane, a PG protease inhibitor, singular protease inhibitor called argenistat. And what was most important, even though the study did not meet its primary endpoints due to some hepatotoxicity and recruiting of some arms that were not as relevant for measuring the subclinical groups. What was amazing is that only the group that had P. gingivalis DNA detected in the saliva at baseline, they showed a dose response to this gingipane inhibitors, which was a portion of the protein toxin that I just showed you, with the 50% slowing of cognitive decline as measured by the ADS-Cog11 test at 80 milligrams, and then 42% slowing in the 40 milligrams. And so these reductions were seen across the board of both 24 and 48 weeks, and multiple correlations that were done as the ADS-Cog11 test, the clinical dementia rating, sums of boxes test, the mini mental state exam, and the ADS-ADL testing. So I'd like to share with you a little bit of what we have in time remaining on the development of a specific biotherapeutic against this bacteria. It was derived actually quite a few years ago as a whole cell bacterial extract, and at the time, which was considered sort of standard development for foreign antigens and mouse monoclonals. Ultimately, it was down selected for its breadth of binding on multiple clinical strains. We picked it up and did some additional work on it, and we're able to demonstrate by building a large PG family genotyping landscape, looking at many 105 clinical isolates. Here you see to the right, these arrows are in fact where we had sampled and tested across the family of PG, the genomic family of PG, the ability for this monoclonal to recognize and bind to this larger protein complex, and found those all to be positive. Initial studies suggest that it has a very profound impact on the bacteria being able to maintain its monoclonal biofilm. As you can see down here in the lower left, the decorations and these white spots that you see on a scanning EM are the actual gold labeled monoclonal antibody binding to the budding outer membrane vesicles that I spoke of as these bud off the bacteria by the hundreds and thousands over days, weeks, months, and are released into systemic circulation. Here, if you do a close-up, you can see them avidly binding to the outer membrane vesicles that are purified and released. In addition, these are able to, in a hemagglutinating assay, of course, inhibit that interaction as well. So you've got this multi-complex functional toxic protein that are in these vesicles, providing sources for the bacterial survival, metabolism. The mechanism of action for the monoclonal seems to be interfering very early on in the processing of the hemagglutinin A gingipanes adhesin complex, which the bacteria essentially absolutely requires for its defense and survival, as you can see here. This essentially seems to be turning off and negating the ability of these outer membrane vesicles to provide all of the necessary support functions for the bacteria. This monoclonal has a relatively high affinity nanomolar binding to its target. Most importantly, it's resistant to cleavage or proteolysis by the bacteria's protease system itself, which it uses, of course, to defend itself against human monoclonal antibodies. It has undergone clinical testing in a cGMP-like material that has been produced, found to be safe in dog toxicity studies, has been used in an initial phase 1 trial where it demonstrated in groups that had received subgingival application, where the monoclonal is laid along the gum line at 3 micrograms per tooth, that no bacteria was able to be recovered from those patients at 3, 6, and 9 months, with only one breakthrough patient at 1 year, as compared to controls where all of the patients had already been returned and recolonized by 3 months, 6 months, 9 months, and a year. A second phase 1/2 randomized double placebo controlled study is planned for this biomarker, where we'll be looking at the roles of it on systemic biomarkers of inflammation. And with that, I'd like to thank all of our partners over a number of years in this work, and in particular, individuals listed here from our team across multiple universities and institutions. With that, I'll take any questions. Thank you.
Yes, thank you, Dr. Peter. Any questions from audience? Okay. Fine. If you do not have any questions, then we can move ahead with the next presentation. Dr. Peter,
Okay.
can you stop sharing your screen? The next presentation, Dr. Brandon Luke, is not available. He just messaged yesterday. though he's not able to join. That next move, can we move ahead with Dr. John Hua? Is he available? Another one is Dr. Gabriel. So some of the two of the audience, like one is like Ken, Dr. Ken Ware has joined with the name of organizer at Scholars Conference as Zoom account. So may I know the another person name, because we are unable to identify the persons who are here. Dr. Raquel, is it possible to start presenting yourself?
Yes, sure. My presentation was scheduled for much later, but if you'd like, I can present now, no problem.
Yeah, that sounds great. Can you please share your screen?
Yes, yes. Just give me a second, okay?
Thank you. Yeah, can you put it in presentation mode?
Okay. Can you hear me fine, or should I get some headsets?
No, it's fine. Can you put it in presentation mode?
Yes, yes, of course. So, like this?
Yeah, that sounds good.
Yeah.
Yeah. You
Okay.
can go ahead. Thank you.
Okay, no problem. So, good morning to everyone, and thank you for being here today. Today, I'll be presenting the work that I developed during my master thesis, which was based on an electrochemical microRNA-based biosensor for the diagnosis of Alzheimer's disease. This project was developed in Biomark Sensor Research, and it had a small stay at I3S, both institutes in Portugal, where I'm from. Okay, so I'll start with a little bit of an introduction first and show you why I chose Alzheimer's disease. First, this is a highly prevalent disease that affects around 50 million people worldwide, and it is associated with a very high annual societal costs. In the US alone, it totals over $800 billion per year. The global prevalence is also expected to keep rising and increasing as the population ages and gets larger, and is expected to reach over 135 million people by 2050. This is especially worrisome since there are no curative or preventative treatments available. The treatments available today can only mask the symptoms or delay the appearance. There are a few diagnostic tools available, but they are both expensive and invasive. So there is a high need for more affordable and especially minimally invasive diagnostic tools for Alzheimer's disease. Alzheimer's can be detected through a lot of methods. We chose electrochemical biosensors since they have been appointed as helpful tools in point-of-care diagnosis. They are small, portable, easy to use, low cost, fast responding, very sensitive, and require only small amounts of samples. The sensitivity is especially enhanced when using oligonucleotide-based biosensors, such as the ones based on microRNA. This is due to the high affinity of the target to the probe. In this project, we chose microRNA 34A because of the high amounts of cumulative literature indicating that there is a link between this microRNA and Alzheimer's disease. Firstly, we see that this microRNA can weaken the oxidative stress defense pathways, which have been recently appointed as one of the main causers for Alzheimer's. Then this overexpression of this microRNA has been shown to be done in the brain of mice and was associated with cognitive impairment, tau hyperphosphorylation, and the accumulation of intracellular A-beta, which are well-known hallmarks of Alzheimer's. Thirdly, this microRNA was shown to be upregulated in the brain of early AD patients, and this upregulation is positively correlated to the severity of the AD pathology. Finally, this microRNA can be detected in the blood and CSF in free-floating form, and the expression is augmented in AD patients. All of this makes microRNA 34A a promising new blood biomarker for AD. Moving on to the experimental section, this was divided in 4 tasks. First, the design of the biosensor, then its calibration in serum and in buffer, then the specificity and selectivity tests. In this case, we used 2 interference present in serum and in plasma, microRNA 107 and creatinine. And then an in vitro proof of concept, which was done based on our final application of using the biosensor as a detector of Alzheimer's. We did this using cell culture, a cell line, which is typically associated with Alzheimer's as HSY5Y, which was differentiated to neurons using retinoic acid. And then the stimulation of microRNA 34A was stimulated with 6-hydroxydopamine. We then did some immunocytochemistry to prove that the secretion was augmented and the differentiation was done properly. And then we tested the biosensor using cellular media samples in which our cells were cultured. So the biosensor design is quite simple. It starts with a commercial carbon screen printed electrode, which is then cleaned using sulfuric acid to activate the surface. Then we modified the working electrode with some gold nanoparticles, nanostructures, in order to increase the area of the working electrode, the active area. We then modified the surface with 3-mercaptopropionic acid in order to introduce some carboxylic groups into the surface, which could then bind our amine-terminated probe. The surface was activated through an ETAC NHS reaction, and then the mimetic probe of DNA nature of anti-microRNA 34A was linearized through heat and bound to the surface. This is a chemical link, much stable than an electrostatic link. Then the non-specific binding sites were blocked using asparagine to prevent non-specific binding. And finally, hybridization with a mimetic microRNA 34A was promoted. All of this was followed up by electrochemical analysis using a redox probe. This is a very commonly used probe of iron 2 and iron 3, in our case done in phosphate buffer. The electrochemical techniques we used were also very common, and we, in this case, used cyclic voltammetry, square wave voltammetry, and electrochemical impedance spectroscopy. Okay. So now I'll give you some results. And the first result that we get is a visual one. When depositing the gold nanoparticles, you can see that the working electrode turns from black to gold. We can see through cyclic voltammetry that there is an increase in conductivity after gold deposition. Same can be seen in square wave voltammetry. And in electrochemical impedance spectroscopy, we see that there is a decrease in impedance and a decrease in resistance to charge transfer, which can be seen as the semicircle. All of this indicates a successful modification of the carbon screen printed electrodes with gold nanoparticles on both morphologies. Moving on to the remaining steps of the construction, we can see that as the steps go on, there is an increase in impedance and in resistance to charge transfer. All of this indicates that non-conductive material is being added to the surface, and is a good indication that there is a successful modification of the surface in both morphologies. We then proceeded to a calibration of the sensor in both morphologies to see which one performed the best. This calibration was done in hybridization buffer, but unfortunately on the morphology 2, we were unable to get a linear fit with all 5 standards. So we moved on to a linear fit with only the 4 lowest standards, which gave similar R-squares for both morphologies. However, if you take a close look at the slope, you can see that the slope is much higher on morphology 1, which indicates that this morphology provides a higher accuracy for the biosensor. In order to understand why we went through with SEM of the surface, as you can see here, morphology 1 has a very much higher surface coverage and a rather different shape. It has somewhat star-like shaped, whereas morphology 2, has more of snowflake-like structure. We think that this star-like shape also gives a higher surface to volume ratio of the particle, which diminishes spherical impedance of binding of the probe to the target. So we decided to go on and use morphology 1 for the remainder of the studies, which was done then a calibration in serum. We did this to get more information, and because this was one of the main applications, we believe in serum or in plasma. We did this in filtered and diluted fetal bovine serum in 100:1 ratio. The filtering was done in order to prevent additional interactions, but as you can see here, there's still a tenfold reduction in slope. Although the filtration was useful, you can still understand that serum is much more complex than a hybridization buffer, which can explain why there was a reduction in slope and therefore in accuracy. We then moved on to the specificity and selectivity tests. And as you can see through the graph, there is no major interference with the biosensor for both the substances tested. We moved on to the in vitro proof of concept, and the first results that we get are in bright field. You can see in the images stand out that when introducing retinoic acid, which is the DIF conditions, into the media, we induce a more spread out morphology and nurate-like extensions, which indicate retinoic acid-induced differentiation. When exposing the cells to 6-hydroxydopamine, or 50 micromolar or 100 micromolar, we see cytoplasmatic condensation and nuclear, which can indicate 6-hydroxydopamine-induced apoptosis. More or less the same can be seen in fluorescence microscopy. This was done by staining nuclei with TAPI, by staining the microtubuli with tubulin beta-3 antibody with a green tag, and by staining the actin filaments with phalloidin. We can see that when exposed to retinoic acid, the cells present a higher microtubuli and actin filament organization, which are both cytoplasmatic proteins. We also see dimmer and larger nuclei, which are again indication of retinoic acid-induced differentiation. Then when using 6-hydroxydopamine, especially 100 micromolar, we see a higher proportion of condensed nuclei, which again indicates an induction of apoptosis by this neurotoxicant substance. Uplifted by these results, we decided to test the biosensor with cellular media samples, meaning cellular extracts of the media in which the cells were cultured. As you can see here, we tried 3 different conditions, one with media with only retinoic acid, and then 2 medias supplemented with 6-hydroxydopamine, one at 50 micromolar and one other at 100 micromolar. As you can see here, there is an increase in resistance to charge transfer shift as the concentration of 6-hydroxydopamine in the medium increases. This means that we were able to stimulate the microRNA 34A secretion using 6-hydroxydopamine, and this secretion seems to be concentration dependent. More importantly, we can see that our biosensor is able to detect microRNA 34A in biological samples. So I'll give you a roundup of the main conclusions. First one is that our design can detect microRNA 34A in buffer, in serum, and in cellular media. The second is that the morphology 1 shows a greater improvement of the biosensor's performance, perhaps due to its higher surface coverage and rounder appearance. The third one is that the biosensor has a linear detection range of 100 picomolar to 1 micromolar, both in buffer and in serum, which is similar to other microRNA 34A biosensors described in the literature. Then we saw that the differentiation and stimulation protocols implemented that were rather new were successful. And then finally, we saw that the biosensor can identify microRNA 34A in biological samples, which is highly optimistic for the application of this microRNA-based biosensor in biological samples in the future. In the future, we would like to experiment with different goal morphologies and deposition techniques. We would like to optimize the hybridization performance of the biosensor in order to lower its limit of detection. We would like to perform more stringent specificity and selectivity tests to be able to say with a much higher confidence that our biosensor can only detect microRNA 34A. We would like to evaluate the stability of the biosensor to see for how long we can hold on to this biosensor before it becomes unusable. And if we ever go to clinical trials, we will have to test the biosensor in human plasma, both of AD patients and healthy controls. So in a nutshell, our work came from the market need for new, less invasive and early stage diagnostic tools for Alzheimer's, which led us to the idea of developing a microRNA-based electrochemical biosensor for the diagnosis of this disease. The design that we came up with was a gold-modified anti-microRNA 34A-based carbon screen printed electrode, which showed great results, being able to detect microRNA 34A in buffer, in serum, and biological samples. And we hope that the work presented here today will lead eventually to the introduction of a cheaper and less invasive AD diagnostic tool that will facilitate the access of many patients to a diagnosis and to timely care. So now I would like to thank first the organization for letting me be here today. I would like to thank all of you for listening to my presentation. I would like to thank my supervisors, Sofia and Felizmina, for supporting me throughout my master thesis. And I would like to give a big thank you to Biomark, which is the institute which I'm currently working on, for sponsoring my master thesis presentation, for supporting my stay here today, and for continuing to support my work. And that's it. If you have any questions, feel free to ask them.
Thank you, doctor. Okay.
Yes.
Then we
Thank you.
can go ahead with the next presentation. There are no questions.
Yeah, I don't think so. Yeah,
Yeah,
you
that's
can move
fine.
on. No problem.
That's fine. So the next presentation is Dr. Gabriel Saratsky. We'll wait for another 5 minutes. Please be with us. But fine, like we do not have the next speakers availability, and we are actually following up them. And we have a video presentation, recorded video presentation by Dr. Rafael Pila. I'll share the screen. It's a recorded video, so you can listen to his voice as well. Okay. Thank you.
Yeah, sorry for interrupting. I don't know if it's just my problem, but I cannot hear anything out of the presentation. I'm aware that we should be hearing something, right?
Yeah, I can't hear too.
Yeah. Okay.
Oh, just a minute. Huh? Yes, you're unable to hear the voice of the Dr. Rafael because it's a recorded presentation. I can able to hear.
Yeah, we can hear your voice, but not the recording of Dr. Raphael. Yeah,
Okay,
that's
just
the
a
problem.
moment. Okay, I'm sharing the presentation in chat box. Just have a look. And I will try to share over here again. Can you able to hear now?
No, we can't hear.
Yeah,
But
me
you can
neither.
see.
Yeah, we can see.
I think you should change the audio share from the application.
Can you hear me now?
We can hear you, but we can't hear the video.
Can you hear now?
Just a second.
Dr. Rafael, can you hear now?
At the moment, I'm still seeing your screen, like the files. No file is playing.
Just find them. Dr. Gabriel, can you hear me?
Yes.
Yeah, can you start sharing your screen, please?
Do you mean Zaretsky?
Yes.
Okay.
Zaretsky.
Yeah, sure. I can.
Thank you.
Hang on, share screen. Yep. Should I start presentation or?
Yeah, you can start.
Okay, fine. Okay. So good morning, everybody. I'm going to talk. Sorry?
Thank you. Thank you.
Okay. Sorry?
Thank you. You can go ahead.
Yes. Okay. So I'm going to talk today about a mouse model of Parkinson's disease, where we applied telomerase activators and found an improvement in motor function and also alpha-synuclein pathology. Okay, so for some reason, okay, yeah. Okay, so for those of you who are not so familiar with telomerase, normally in its canonical function, telomerase is a reverse transcriptase consisting of a catalytic subunit shown here in green and an inherent RNA component, which also contains the template for telomere synthesis. However, the whole of my talk, I will talk about non-canonical functions of the TERT protein, which I can also talk, is also called telomerase. It's a bit confusing. Yeah. Okay, so what we found long time ago is when we overexpress the TERT protein, for example, in normal human fibroblasts, which don't have telomerase, usually it's located in the nucleus, shown here with a red or pink signal. However, if we apply either acute or physiological oxidative stress, like hyperoxia, which is 40% oxygen in the incubator instead of 20%, you can see that the protein leaves either partially or completely the nucleus. It takes much longer with the mild chronic hyperoxia. And if we then co-stain with a green signal for mitochondria, then you can see a lot of overlap in the form of a yellow signal. That means the TERT protein shuttled from the nucleus partially to the mitochondria upon oxidative stress. And functionally, it results in lower reactive oxygen species already under normoxia, but more under hypoxia. So red are the H-TERT overexpressing cells. And if you look onto the mitochondrial DNA damage, it's also decreased under H2O2 and under hyperoxia. And importantly, this TERT localization in the mitochondria is also associated with a much lower resistance against apoptosis. For example, again, if you apply either H2O2, hydrogen peroxide, or etoposide, which is used as a cancer therapeutic for breast cancer. So, however, this was just where we started from. We were then interested whether TERT has also a non-canonical function in brain in vivo. And we looked into cultured primary mouse neurons. And during the culture, after 2 weeks, they have lost telomerase activity. So this is activity now. In the mouse brain, knockout mice don't have it, of course. It's still there postnatally, but it decreases within the first 3 weeks. And at 2 months of age, there is no measurable telomerase activity in the mouse brain. If we now look into human brains, it's slightly different, also meaning that telomere and telomerase biology are slightly different in rodents and in humans. So these are very early developmental stages, and the weeks are post-conception. So you can see post-conception week 6, there's still a lot of telomerase activity, but it goes dramatically down. And by roughly 3 weeks, 20 weeks, there's hardly any telomerase activity, and that persists in adults. So this all has been published. However, what we found when we look into human hippocampal tissue, looking for the different CA regions, either with immunohistochemistry or immunofluorescence, where TERT is shown in green, you can see that neurons have a lot of TERT protein. There is a little bit in microglia, which is not surprising because they are macrophage derivatives. However, there's no TERT protein within astrocytes. Now, here the antibodies of TERT are shown in red. Similarly, when we look to cultured mouse neurons, we see a lot of TERT in the neurons and microglia, but again, nothing in the astrocytes, and that's just a control that the antibody is specific. So from there, we progress to look into 2 different neurodegenerative diseases. So this is Alzheimer's disease and also a mouse model of Parkinson's disease. And that's what I will talk in the remain of my presentation. So what we published a long time ago when we analyzed human Alzheimer's brains of different Bragg stages, our initial hypothesis was that there is a decrease with the disease. However, we didn't find this. I don't show it here. But what we found is similarly to what I presented before for the cell cultures, that we see more TERT localized within the mitochondria in CA1, CA2, CA3, predominantly in Bragg stage 6. And if we again do a brain staining, TERT protein is stained in red in controls, while higher Bragg stages, you can still see the red staining, but of course we have neurofibrillary tangles of tau protein, which you can see here in green. And what we found is that there's never a co-staining between the pathological tau and the TERT, which can mean that either TERT is displaced or those neurons which have high amounts of TERT don't develop neurofibrillary tangles. However, in order to do a more mechanistical study, we again used cultured neurons from the wild type mice shown in blue and TERT knockout mice shown in red. And what we found is that the knockouts have much higher levels of oxidative stress in the dendrites. And we also looked for lipid peroxidation products, and we found that there's a higher lipid peroxidation in the soma. So clearly, if you have TERT protein, like in the wild types, it is protective against transduced mutated tau. So we used the P301L mutation form. So we were aware at this time already that there are different telomerase activators available, some of them freely on the market. So you can see here the plant extract from Mongolian milkveg called TA65. It is so it's a plant product, but you can also have the small molecular derivative of cycloestrogenol. It's called GN 510, and we used both of them in our experiments. So first, what we did is we treated for 3 months, 2-year-old, so quite old mice. And what we found in a different study is that in mouse brains, TERT expression decreases with age, while when we treat those old mice with, which were around here, so 2 years of age, with both activators, then we have a significant increase in TERT expression. If we now look for behavioral outcomes, we used a Steiter rod because these old mice are not very well on any other moving rod. So we have here young mice which can stay for 1 minute easily on this rod. However, the old ones and the controls are always treated with the Solo and DMSO. You can see they can only stay very short time. However, when we treat them with both activators, at least for the GRN 510, it's highly significant and almost to the level of the young one. The TA65, we didn't have so many mice. However, in order to be sure that it's really neurons which are expressing TERT, because of course in the brain itself we have different cell types, we cultured again wild type neurons, treated them with either GRN or with TA65, and with the highest concentrations we applied, we can see a strong induction of TERT expression. So yeah, probably to this audience don't have to explain about Parkinson's disease, just to quickly remind you that Lewy bodies, and which are alpha-synuclein aggregates, are the main feature of brain pathology. It leads to the loss and decrease of dopaminergic neurons and their function. And you can see this on the phenotypical level as movement disorders, which are tremor, Parkinson, so all different kinds. And it's known on the molecular side that there are also mitochondrial and autophagy dysfunctions involved. And we will look more closely to these. So what we used then was a transgenic mouse model, which was generated by Elisée Maslaya 20 years ago. And it expresses wild-type human alpha-synuclein. And you can see in the brain, you have almost the same alpha-synuclein inclusions like Lewy bodies. Interestingly, when you look for the kinetics of the transgene, it takes quite some time to increase in the hippocampus and also in the striatum. And in my view, this just means that it is an excellent model for age-related neurodegenerative diseases, which are normally don't occur in young subjects. So this is a quite good observation. And to characterize PD more closely, dopamine levels were characterized, which are down, and also tyrosine hydroxylase activity was down. in these mice. So they are quite a good model for Parkinson's disease and have been used for many different types of experiments by these authors and others. So what we did is we started at 4 months of age and treated the mice for 14 months. During this time, we did different motor balance analysis. For example, first of all, we checked after an 18-month-old mouse brains, and if we could, we discriminated between males and females, and you will see in the next slide why. So in the females, both activators increased the TERT expression significantly. In males, there was a similar trend, but GRN was significant, and if we pooled both sexes, both activators were strongly significant. So now we come to the motor balance activities. So it's a rotor rod test, which measures speed, time on rod, and the distance traveled. And interestingly, we found a sex-specific response. So in females, only the plant extract TA65 showed a significant improvement of the behavior or the motor balance, while in the males, it was only the GRN 510. And interpretation of these results we concluded was that male and female brains are not the same. And I'm not a Parkinson's specialist, but I know that there are differences in gene expression and also in other parameters. So it's not completely out of the blue that we see such difference. However, in other parameters, we not necessarily saw them. So we looked into walking speed, which is a readout for human bradykinesia. However, again, we found in females a strong improvement with TA65, but only a trend with the GRN 510, while in males this time, both activators were significantly improving the walking speed, which was slower in the males in the first place than females, perhaps due to the fact that the males were much fatter and they're slower. So we did a stride length test, which is very similar to human gait test, which is normally done with electrodes where the patients walk on. We did a very low maintenance analysis type where we painted the hind legs. The right side was blue and the left side was red. And you can already see that the controls are quite wobbly. So the stride length is very variable and it's also quite short. While when we treat the same age animals with both activators, you can see you have quite a long stride and a much more even stride length. And this is the quantification of it for the left and the right leg from the females. You see a significant improvement in the size, in the length of the stride, and most importantly, the variation between the strides, which is very heterogeneous here, is much more homogeneous in this respect. And in this case, and now I show you the results for the males, we didn't find really any differences. So this test was not sex specific, but both gender or both sexes significantly improved their stride length as well as their variation between strides upon treatment with both telomerase activators. So in order to do some cognition test, normally we would use the Barnes maze test. However, due to the locomotion problems we saw before, we resorted to a novel object recognition, where first the mouse is presented with two equal objects and then switch to the same old and a new object. And you would expect that the mouse has more interest and curiosity in the new object. And while this is not the case in the controls, the females improved their recognition of the new object with both activators. However, in the males, again, it was very strongly the GRN 510, but no effect of the plant extract TA65. So what I told you previously, we know that TERT protein within the mitochondria is able to improve aspects of respiration and to decrease ROS release. And we did this with an amplex rat test here where we isolated mitochondria. This time we pooled the sexes and stimulated them with different substrates, pyruvate malate and rotenone and complex 2, substrate succinate and rotenone, in order to see whether there is a decrease in the release of ROS. And indeed, we find this decrease with TA65 in the forward and the reverse electron flow, but we didn't see any effects with GRN. So that means the effects we saw with both activators previously in the behavior tests are probably not due to, or not due entirely to, the ROS decrease of ROS generation and release. So we looked for different mechanisms. So after we sacrificed the mice, we looked into alpha-synuclein pathology, and here you see images of the now let me get rid of this. Of the CA1 hippocampal area, and all of these images are taken with the same exposure. In red, I show the phosphorylated alpha-synuclein, while in green is total alpha-synuclein, and here they are merged. And you can already, by looking into these images, appreciate that there is a decrease with GRN 510 and almost a disappearance of the phosphorylated and the total alpha-synuclein after 14 months of treatment. So this is the quantification where we analyze CA1, CA3, and neocortex, and the total alpha-synuclein levels decreased with both activators in all regions. The phosphorylated was mostly significant in CA1 with both activators. For CA3, mainly with TA, because we had the outlier for GRN. And in the neocortex, it was also predominantly TA65, although you can see that there is a clear trend also with GRN 510. Then we looked into the ratio between phosphorylated and total, because considering that already the total level is decreased, we wanted to see whether there is a specific decrease of phosphorylated alpha-synuclein, and we found this. My mouse here. Where's my mouse here? So with TA65 again in CA1 region and also in the neocortex, and just a small trend in CA3. So there is a variability within brain region, but we also didn't have enormous amounts of animals, so it could have been that with more we would have been able to see more, but we had very little funding, so we had to reside to these numbers. In order to exclude that any of those effects obtained with immunofluorescence are artifacts, we used also immunohistochemistry and a different primary antibody for the total alpha-synuclein. And here we only analyzed the layer of pyramidal neurons, because you can see the other layers are very homogeneous. And I think you can already appreciate from looking onto those images that the numbers of the pyramidal neuron bodies in this layer is decreased dramatically, and we quantified this here. And we did analyze exactly the same region and layer with immune fluorescence, and you can see that both telomerase activators decrease the amount of alpha-synuclein in these neuronal bodies. And there's a very good concordance between the immunohistochemistry and the immunofluorescence. So that means it's true and not kind of methodological artifact. But of course, most important for the formation of Lewy bodies is aggregated alpha-synuclein, where we used an antibody here shown in red, and this time it's the neocortex. And again, total alpha-synuclein is in green, and you can see both of those components decrease significantly with the treatment. Again, same exposure. You can see here in the controls the merging between the aggregated alpha-synuclein and total alpha-synuclein in yellow signals, and the quantification in CA1, it's a significant decrease with TA65, but a trend with the GRN. In CA3, both activators give a strong decrease, almost 40%, of decrease in aggregated alpha-synuclein and a strong significance as well in the neocortex. So to finish off, as I said, that the mitochondria are probably not the most important mechanism. So we looked into autophagy, although this are more or less pilot data. We analyzed the p62 adapter protein and the LC3B protein, which is localized on the autophagosomal membranes, and a decrease of those proteins means that there is more autophagy. So for the LCP3, it means autophagosomes are more quickly turned over. And you can see for the LC3, there is a significant decrease in the CA1 with both activators. There's a trend in the CA3 and a slight trend in the neocortex. And similarly with the P62, there's a significance in the CA1 region, a strong significance with TA in the CA3, and just a trend for the neocortical region. So my summary is the following. I hope I could convince you that a treatment with telomerase activators improved movement, balance, as well as gait in a transgenic mouse model of alpha-synuclein or of PD, so using human alpha-synuclein expression, and the treatment of with the two telomerase activators also importantly decreased the total, the phosphorylated as well as aggregated alpha-synuclein. And we think that the main mechanism is not the mitochondrial localization, which we couldn't directly analyze in this model because the antibody, the TERT antibodies available are mainly for human and for cultured mouse cells, but don't work in mouse brains. So we think that TERT activates rather a protein degradation mechanism such as autophagy, and other studies before have shown this, for example, in cell models, and also that TERT is able to be in a complex with, for example, the autophagy protein or the autophagy-related protein mTOR. And here's my acknowledgement. So most of the experiments were done by my then Chinese PhD student, Teng-Fai Wan, who is very talented and very organized. Another master student, Amavir, was involved, previous members of the group. And I also grateful to TA Science, which gave us the two activators for free. Victor Krotschuk is a specialist in autophagy, and Johannes and Mary are from our neuropathology lab. So I thank you for listening to my talk and happy to take questions.
Yes, thank you, Dr. Saretsky. Any questions from audience? If there are no questions, then we can move ahead with the next presentation. Now I invite Dr. Priyanka Chandolia.
Yes, sir. Good afternoon. Am I audible?
Yes. Can you start sharing your screen, please?
Sure, sir. Sure.
We can hear you.
Share your screen, please.
Yes, sir. I'm just sharing my screen. Is it visible, sir?
No. Yeah, Priyanka. Is there any issue?
Just hold on, sir. Actually, there is a network issue. I'm just trying to connect. Is my screen visible, sir? Is the screen visible, sir?
Yes, it is. We can see it.
Okay.
Yeah, that's fine.
A very good afternoon to each one and all. My name is Priyanka Chandolia and I'm currently working as an assistant professor in NIMS Institute of Pharmacy, Jaipur, Rajasthan, India. The title of my topic of the today's discussion is Herbal Remedies for the Cerebral Ischemia and the Other Neurological Issues. So if we talk about the cerebral ischemia, basically it's a common acute brain injury that results in the impaired blood flow to the brain. And if it is untreated, it may result as a death of a patient. And basically it can also result in the cerebral infarctions or the global hypoxia ischemic encephalopathy. So if we talk about the cerebral ischemia, basically we have two types of cerebral ischemia. First one is the global and the second one is the focal one. In global cerebral ischemia, basically it results, and the most common cause of this global ischemia is due to the systematic hypotension. And in contrast, if we talk about the focal brain ischemia, it is the most commonly occur in the brain, and it is due to the obstruction in the arterial blood flow, which stops the flow of the blood or the oxygen inside the brain. Next, we will be including with the sign and symptoms of the brain ischemia. So if we discuss about the sign and symptoms, the sign and symptoms include the face drooping, legs and arm weakness, slurred speech, loss of balance, trouble seeing, and the severe headache. So if we conclude with the pathophysiology of the cerebral ischemia, it includes the vesicular occlusion, which decreases the cerebral blood flow inside the brain, and it also decreases the oxygen demand and the glucose demand in the brain, which will be resulting the alteration in the cellular metabolism, energy producing processes, which will be leading to the alteration in the cell membranes, and that results with the necrosis, apoptosis, which leads to the neuroinflammation in the brain in the cerebral ischemia diseases. Now, our main topic, that the herbal remedies to treat the cerebral ischemia. So there are lots of herbal remedies, lots of natural remedies which can treat the cerebral ischemia, and we can open that we can make it sure that there will be the proper supply of oxygen and the blood flow inside the brain. So here is the first herbal remedy, that is known as Artemisia absinthium. So basically, this is the most commonly known as wormwood, and it is an aromatic herb which includes with the fibrous roots. And basically, if we talk about the properties, it has several properties, such as it can act as an antiseptic, it can act as an antispasmodic, or it can be acting as a cardiac stimulant, which can help to restore the mental functions and the inflammation of the liver and to improve the memory. And if we talk about it, also exhibit the neuroprotection evidence in the reduction of the infarct volume and the lipid peroxidation, which is helpful to restore the antioxidants inside the brain. And if we talk about the focal, which induce the increase in the lipid peroxidation and the administration of this herbal extract before the focal cerebral ischemia, it markedly decrease the ischemia and the reperfusion, which will be leading to the level of thiobarbituric reactive substances in the brain. So if we can see here, this is the diagram which the Artemisia abstinensia plant looks like. Next herbal remedy is Osmimum basilicum. So basically this is also known as the sweet basil. And if we talk about this, this is cultivated in the Asia, in the Africa, in the South America, and in the Mediterranean region. And it is also having the further properties and the activities, and it is used in the treatment of anxiety such as headaches, nerve pain, diabetes, digestive disorders, migraines. So these are the lots of variety of properties of this herbal plant which can be used in the neurodegenerative disorders. And if we talk about the particular neuroprotective effect of this particular herbal plant, it has been evaluated by using the global cerebral ischemia through the reperfusion of the rat animal model. And it also exhibits the neuroprotection with the reduction in the infarction and the lipid peroxidation, as well as restoration of the healthful antioxidants substances. And if these antioxidants are overproduced, they are also helpful to detoxify all the antioxidants. For example, like glutathione, it is acting as the central component of the antioxidant defenses of the cells. Next herbal remedy is that is the ginkgo biloba. Now this ginkgo biloba, basically we use the extract of this plant, and it mainly and basically contains the flavonoids that may be contributed to the ginkgo antioxidants, or it may be helpful for the free radical scavenging effects. And these extract reduces the cell membrane lipid peroxidation in the experimental animal model of the spinal cord injury also, and in the methylprednisolone. It also reduces the bromothrin-induced cerebral lipid peroxidation in the edema. And this extract, it protects the brain neurons against the oxidative stress, which will be induced by the peroxidation, and also decrease the neuronal injuries followed by the brain ischemia. Next, if we talk about the other further activity of this ginkgo biloba, it also exhibits the dose dependently synthesis of the nitric oxide, which will be helpful for the inhibition of the inducible nitric oxide synthase, and cerebral edema, which can increase the intracranial pressure and exacerbates the vascular dysfunctioning, impairing in the cerebral blood flow. Next compound of the herbal compound, which is helpful to treat the cerebral ischemia, is Camellia sinensis. So basically this Camellia sinensis is we include with the green tea, as we lot of people we consume with the green tea. Basically these green tea leaves, it has the minimal oxidation during the processes, and fresh tea leaves is the usually rich in the flavonoid group of the polyphenols and basically these green tea contains the catechins as the active ingredient. So these catechins, basically these are helpful for the provide with the good antioxidant inside the brain, and the hydrogen peroxide level of the brain, which can be increased by the ischemia and the reperfusion. If we give the 0.5% of the green tea extract pre-treatment for the 3 days, it significantly reduces the increased level of hydrogen peroxide, and it also helpful to inhibit the increased production of the lipid peroxidation products. And next, if we conclude with the green tea active ingredient, it also contains the eicosanoid, and this eicosanoid, it is useful for the significantly elevated in the hemisphere by the ischemia reperfusion compared with the another one. So these concentration was significantly reduced by if we give the 0.5% of the extract of the green tea, which will be as a pre-treatment for the 3 weeks. Next ingredient that is known as the bisalin and the jasminodin. So these are the two major compounds that is, they are also known as the Chinese herbs. And these Chinese herbs, these are markedly decrease the infarction area in the rat brain. If we talk about the basilin, basically it shows the ability which interferes with the activities of the N-methyl D-aspartate NMDA receptor plus with the nitric oxide synthase. And if we talk about the next ingredient, that is jasminodin, it is found and it is found to be significantly reduce the expression of the monocyte. Expression of the monocyte chemo attached, that is protein 1 in the ischemia brain in the rats. And they both increase the BDNF level in the mRNA expression also in the hippocampus region in the rat brain, which undergo with the focal cerebral ischemia reperfusion injury. So both these bacillin and jasminodin, they found very effectively to decrease the caspase 3 mRNA expression. So if I last slide, it is the conclusion. So if I conclude with my talk is that the medicinal plants or the herbal plants, they play the very important role in the treatment of the many incurable disease such as neurological disease and the brain ischemia or the reperfusion. It's a complex progressive injury which involves the mechanisms such as the survival, apoptosis, mitochondrial dysfunction, calcium overload, inflammation. So all these, etc. So all these are the basically the mechanism which involve in this cerebral ischemia injury. So it is difficult to describe the mechanism of the protective agents against the ischemia and the reperfusion. In conclusion, these development of the protective agents from the traditional herb medicine is a promises direction inside the treatment of the ischemic cerebral injury, which will be related to the neurodegenerative disease. And in future, if I talk about like more attention should be paid to the natural and herbal compound so that it can transverse the triple B, that is the blood-brain barrier, and it can be having the wide therapeutic time windows, which will be clearing out the pharmacological trends and with the fewer side effects. So at the last, I would like to thank you all. This was all about my topic. If any questions, you may please ask.
Thank you, Priyanka.
Thank you so much
Is
for
there
the
any
time.
questions from our? Is there any questions from the audience?
Any questions?
Okay, if there are no questions, then we can have a break for 10 minutes now. Then we can again continue the presentations. Thank you.
Okay, sir. Thank you so much, sir. Thanks a lot.
Hello.
Hi, welcome back. Dr. John,
Yes.
don't know how. Can you share your screen, please? We're actually waiting for your presentation. Like we have moved ahead with the next presentations and we give break. Now you can start your presentation.
I can start?
Yeah. Can you share your screen?
Yes. Okay. Let me get started. Thank you.
Yeah. Yeah, thank you.
I thought, because I thought my presentation is 11 o'clock GMT. So it's 7 o'clock here, my local time. So let me get started. Thank you.
Yeah, thank you.
All right. So let me start my slides. And then share. Oops. Share screen. This is my Zoom. Share screen. That's my slides. All right. Can you see all right?
Yes. You can go ahead.
Okay, great. Thank you so much. So I'm going to talk about differential. Thank you. I'm going to talk about differential laminar activation, dissociates encoding and retrieval in the human medial and lateral entorhinal cortex. Next slide. So I am a faculty working at the FM Kirby Research Center for Functional Brain Imaging at Johns Hopkins and Kennedy Krieger Institute in Baltimore, in the United States. So this is we are this is the center. We are a national imaging center in the U.S. So we do mainly research, brain imaging research, usually mainly functional MRI. So we have 3 Tesla MR machine for research only, and then across street we have this so-called high field MR machine, 7 Tesla, to get a better signal for MR imaging. And we also have a 11.7 Tesla preclinical scanner for animal studies. And we have about more than 100 users, from the local and national community. So for myself, my training is in biomedical engineering. I'm currently a associate professor in the radiology department. So the focus of my research in my lab is to develop new MR methods to image brain function and small vessels and the metabolism. Because I work at Johns Hopkins, so over the years, so we had we are very fortunate to have lots of clinicians who are interested in using our methods to look at brain changes in different diseases. So today, what I'm going to talk about is a project we did on the high field 7 Tesla machine to use very high spatial resolution function MRI to look at brain function in the memory system in humans. So this is our goal is we're trying to see if functional MRI, if we can use functional MRI to distinguish the different layers, the functional activation in different layers in the entorhinal cortex, the memory system, which is very important. This is our collaborator, Dr. Becker, who is a neuroscientist, also an expert for functional imaging. So basically what we were looking at is this is the brain circuits we're looking at. So this is the entorhinal cortex. So it has 6 layers. The superficial layers, layers 1 to 3, and deep layers, layers 4 to 6. So the superficial layers are considered an input layer to the hippocampus region, which is more associated with encoding of information. And the deep layers is considered output layer from the hippocampus region, which are more associated with decoding or retrieval of information. So, and this has been studied, previously in lots of literature. So, our goal is we want to design some sort of functional task to induce functional activity in the the entorhinal cortex. So, and then we want to contrast the encoding and the retrieval of information. We expect to see if we have spatial sufficient spatial resolution to disassociate superficial from deep layer. We expect to see a functional MRI signal that's more correlated in response to encoding in the superficial layer, and in the deep layer, it's more correlated associated with a retrieval of information. So this is our design. And first of all, we need to find where the entorhinal cortex is. So this is a whole brain, so-called whole brain high resolution functional MRI, structural MRI image. And this is where the entorhinal cortex is. This is so the entorhinal cortex can be separated into medial and lateral. And these are so-called deep layers, and these are the so-called superficial layers. And then we have an even higher resolution T2 weighted image where we manually segment the medial and temporal, medial and lateral entorhinal cortex, like this. So, and then we start to do functional MRI. There are lots of challenges. It's not trivial to do that, especially in this region. So there are several technical challenges. One is as you notice, the shape of the entorhinal cortex is irregular, so we have to do manual segmentation, and also we have to, in order to get different layers, we have to make our voxel the same dimension, all the same size on all dimensions. Otherwise, we cannot because if we make one dimension larger than the other, then we cannot separate evenly. And the other thing is, it's well noted in the field that because this region is very close to the cavity in the brain, which means there's air in this region, and this is brain tissue. And this is well known that functional MRI, when we acquire with a very high speed, there will be some signal dropouts because of at the air tissue boundary, which obviously affects the signal a lot in this region, so it becomes very difficult. So to do that, we use the method that's developed in our group to reduce signal dropout in these regions. So, for example, this is one example. So this is the images we acquire our 7 Tesla machine. So this is structural image. It looks great. And these are two different conventional functional MRI methods where you can see the signal dropouts in these regions close to air cavities. So, and also this is a problem, for many other diseases. For example, if a subject has a resection in the brain, or has bleeding, you have dropouts as well. And then for subjects wearing, have metal implants, and this is also a problem. So we developed that method to reduce the signal dropout. So this is a method that we call the T2 prep functional MRI. So basically, we use the T2 preparation to get the both signal, and then we use a readout that's very similar to structural imaging to reduce these kind of artifacts. So this is coming back to the example. So this is a T2 prep both images. As you can see, the dropouts in these regions have been much reduced. So, and then we test that in a subject wearing a metallic brace, and the effects are much reduced as well, the artifacts. So, and then we decided to use this in our project looking at entorhinal cortex. So this is one example. This is an anatomical image. There's no distortion. There's the entorhinal cortex. With a conventional method, you see lots of dropouts. But with T2 prep, you see a signal, much better. So this is the 3D view. 3-planar view. So this is and also more importantly, we want to make sure that the temporal signal to noise ratio improved as well. So to measure that, we compare the T2 prep method with the EPI method. As you can see, the T2 prep method has better signal to noise ratio across the layers. And also, more importantly, the signal to noise ratio seems to be changing from superficial to deep layers with a conventional method, but with T2 prep, it's more consistent across layers, which is important. So, and of course, the T2 prep method is, in terms of contrast, it's a T2 weighted contrast. So the signal change is smaller than the conventional gradient echo. But in this case, we still decide to use that because it gives us better signal. Okay, so I try to speed up, through the methods part, and then we can get to more interesting neuroscience part for the audience. So this is the design of the functional paradigm we use. So we basically, the subject gonna see many color photographs. So they're gonna see a photo every 3 seconds. So what they need to do is they need to identify, for example, these are so-called identical pictures. And we call that repeat pairs. These are so-called similar pictures. So both apples, but slightly different apples. And for the rest, they are all not relevant, unrelated. We call the foils. So the pictures, for the pictures, the subject need to identify. They have a button box, so they need to identify whether this is old, new, or similar picture. So for contrast, if a subject successfully identify a repeated pair, so this, and then the first presentation is considered a successful encoding trial. And the second presentation here is considered a successful retrieval. So this is the same for similar pictures. Okay, so these are the imaging parameters we use. I just want to point out this is the T2 prep method we use. So it's a long task, about 40 minutes, but we separate it into 8 runs so the subjects can have a little rest in between. And we use a sub-millimeter isotropic voxel for this study. So this is the main result. So this is so-called profile, laminar profile. So the x-axis are the layers. This is a CSF boundary. This is a white matter boundary. So this is these are considered superficial layers. These are considered deep layers. And this is the functional MRI signal change presented as beta values. So you can see in the superficial layers, they respond more to the encoding activity, and in the deep layers, as we expected it, they respond more to the retrieval of information. Okay, and then we start to look at additional conditions. Not just encoding versus retrieval. So this is so first of all, we'll look at the successful encoding versus the foil condition. So first of all, when we look at encoding versus foil, we didn't seem to see much difference. So we saw that perhaps encoding and the foil are similar in terms of memory response. And then we look at successful retrieval versus foil. Interestingly, we saw some response in the deep layers. That's more response to the lateral. Retrieval of information. Okay. And then when we look at the two different conditions separately, they seem to show some difference. So it looks like retrieval, when you're trying to retrieve information, it's different from a random foil design. So So, okay, so that's, I try to be short and brief, but this is the main results I want to present here. So there's basically we talk about laminar function MRI in the entorhinal cortex, and then we look at differential laminar function activity in the entorhinal cortex. Seem to relate to encoding and the retrieval. So the encoding is considered input, and the retrieval is called considered output to the entorhinal cortex. And with our method, we seem to be able to detect robust signal changes in the superficial and deep layers in response to the encoding and retrieval processes. And this is a very exciting work, actually just published in Journal of Neuroscience. And we continue to do that. And I think the more exciting is the next step. We want to look at these changes in different diseases. For example, in Alzheimer's disease, it seems some literature suggests that the encoding process is more affected, especially for the superficial layers and especially the left entorhinal cortex. While in Parkinson's disease, it looks like the retrieval process in the deep layers are more effective. So these are the things we're going to look at with this method in different diseases, and hopefully we can report some interesting results in the coming years. With this, I would like to thank all my collaborators, especially Dr. Becker and Dr. Huber, and my group and founding support. And thank you for having me. I'll be happy to answer questions. Thank you. Kevin, should I leave? I mean, should I mute?
Yeah. Thank you,
Okay.
Dr. John.
Yeah, thank you.
Any questions from the audience? No, if not, then we can go ahead with the next presentation. The next presentation is
great.
Dr. Lama Saad. Lama?
Yes, doctor. Yes.
Yeah. Can you start
I'm ready
sharing
to
your
start.
screen?
Okay. Okay, my presentation on your screen.
Can you just
Okay.
can you just put it in presentation mode? F5.
Okay.
Yeah. Thank you. The stage is yours. You can go ahead.
Okay. First of all, I would like to thank you for my participation in this great event for the neurology field. I'm Dr. Lama Saad Al-Din from October 6 University from Egypt. I'm a lecturer of neuromuscular disorder and it is surgery. Today, I am going to talk about the artificial intelligence and the virtual reality cognitive rehabilitation in stroke patients with unilateral spatial neglect. As we know, before the stroke, any individuals have a good left and right hemispheres. So there is a good upper limb and lower limb functions. But after the stroke, the patient, as we see here in this picture, he will have a lesion in one hemispheres, which lead to ignore to the contralateral lesional stimuli. The visual reality and the artificial intelligence offer promising approaches to the stroke rehabilitation. The artificial intelligence could be in more types. Mainly there is a machine learning types and other methods. The machine learning types have a classical and the neural networks. The classical methods as a supervised learning environment or unsupervised learning environment. While the neural network has a lot of methods like the virtual reality rehabilitation.
Artificial intelligence also indicates that the use of artificial intelligence in the form of information and communication technology, the virtual environment that we use in this study, or the robotics, improves the patient engagement in the movement and stimulate the activity of daily living performance and promote social interaction. So we used for the unilateral special neglect patient the virtual reality environment to engage the patient to be in a virtual environment and to act in this environment as a rehabilitation, especially for the neglected patient that have a special neglect. So we use the virtual reality Xbox 360, which is an advanced method and allows the users to interact with a multi-sensory stimulated environment and receive a real-time feedback on performance, but with a special program for the rehabilitation. Before I start, what is the unilateral special neglect? It is the impairment of the ability to respond to the stimuli in the contralateral lesion space, not only in the body parts, but in the space of the patient. The virtual reality, it's a computer-based technology which received a real-time feedback. However, the kinect methods of the virtual reality, it is advanced method as a motion sensing inputs without any contact as gloves or any wires. So this is the type of virtual reality. And as I said, we use the Xbox camera. The unilateral special neglect after a stroke, it's leading to the patient to act as if whole region of the space contralateral to their damaged brain lesion don't exist anymore. Although there is a visual feedback is normal, but he don't react or don't perform any movement in this affected space. So the patient's unaware of his left side or the affected side. There is many categories of unilateral special neglect at the personal and pre-personal and extra-personal spaces. The personal space, it's means the patient's body parts, and the pre-personal space with the patient dealing with the environment and with his self-hygiene. While the extra-personal space, it is the patient engagement in the external environment. Okay. What's the relationship between the artificial intelligence and the virtual reality? The virtual reality is designed to be more interesting, more enjoyable than the traditional therapy. And also it could give the patient a neural plasticity, or it could stimulate the neural plasticity in the brain by the repetition and the intensity and the training through the neural rehabilitation. So we called it artificial intelligence rehabilitation gaming system, as there is motor and pre-motor systems through these routes and through the virtual representation of the body, which enhancing the observation of the movement the patient could see, and so it's drive the mirror neuron system in the brain. Okay, what is the concept of the plasticity with virtual reality? As a traditional information, we know if you do a touch or pressure on a coin with a lump of clay, it will still put in this clay. So the virtual reality and the learning through the virtual reality, it's like this symbol or like this example. The neural circuits in the brain must reorganize the response and the experience through a multi-sensory virtual stimulation to adapt the patient and to learn the patient, especially the special neglected patient, to learn more about the neglected space. So the virtual reality allows the user to interact with not only the visual environment, but also with virtual objects within this environment. And this enables the therapist to determine the optimal environmental factors for the patient. So it improves the interaction space between the patient and between the virtual environment and also a virtual objects to perform tasks through the neglected space in the patient environment. So the main benefits of the virtual reality, it is a real life situations, immediate feedback, it's give multi-sensory stimulation and repeated learning trials, and it is safe for the patient, and we can change the environment. So it's mainly stimulates the remapping of the space in the brain and also represent a contralesional side of the space. The instrumentation and assessment for our patients, firstly, it is the paper and pencil test of extrapersonal neglection, like the line cancellation task. As we see here, the patient with unilateral special neglect neglected all lines, can't check it in the left side, while checked only the lines in the right side. So there is a neglect in the left side for this patient. Also, the paper and pencil test includes the line bisection test. When we give the patient a piece of paper with lines to check it in the center, but the patient always does his check to the affected side and ignore the neglected side, as we see here and here. Okay. Also, the clock drawing test. The clock drawing test depends on, we give the patient circle and put on then the numbers of the clock. As we see here, the patient put all numbers to one side and neglected it in the other side. Also, we have a measurement of the self-awareness of the neglection by the severity of the Catherine-Bergego scale, which have a severity from 0, no neglection, to 3, the severe neglection. Okay. Let's start the intervention procedures. The virtual reality group is a study group which receives both the virtual reality and also the conventional treatment. The virtual reality, which is a type of the artificial intelligence, we use it for the patient, but before the intervention, we should do a preparation before the intervention for the cognitive function of the patient to be more mentally relaxed. Okay, here there is a example for the patient with a stroke patient. As we see the patient in front of a projector, and also the projector gives him the appearance of a virtual environment on the wall. We can use this program with different environment. We can use it to do a movement with his upper limb or to do movement with his lower limb. Okay, in this program, the patient perform movement for his upper limb. Also, there is another program. The patient do movement here, but also the patient do movement in with the upper limb. But the control group mainly received the movement and scanning techniques, the reading and the copying training, the limb activation training, and the copying line drawing on a dot matrix. It is a conventional treatment program and the traditional program, which is called a behavioral rehabilitation methods for the unilateral spatial neglect. The movement and scanning techniques like this, the patient move his limb, but firstly he used the visual feedback and to move his limb, like in this picture, and also the limb activation training. The patient firstly look to his affected limb with open eye, and then the patient perform the movement many times. Before we talk about the results, I want to clarify points. We can use many different programs for the virtual reality, and we have here we used 3 or 2 main programs for the upper limb movement, and it's very it was very interesting for the patient, and the patient feel he is interesting and wanted to move more and more and more. But also it depends on the patient. There is patient need to move his limbs more than the upper limb, or the patient need for balance rehabilitation. So we can use different visual environment and virtual objects according to the patient's needs. Again, in these results, in these slides, we show the results, the comparison between the results of both groups, pre and post treatment. We can see that the study group in the blue color, and the group B, the conventional group or the control group, in the red color. We show these results show that there is improvement in the line cancellation test for the study group more than the control group. Also, the clocked and wrong levels, the line by section test, and the Catherine-Bergigo scale. The improvement for the study group was more than the control group, but also the control group have a percentage of improvement. When we assessed also the visual reality assessment program that we used, we show that the virtual reality repetitions, the number of repetition for the study group, it was more after treatment, post-treatment, it was more for the study group, more than the control group. And also the movement distance for the study group, it is more, have more improvement than the control group. Also, the study shows that there is a correlation between the line cancellation task and the Catherine-Bergigo scale, and also with the visual reality rehabilitation, the visual reality assessment. That means we could also use the visual reality assessment to get a more objective data for the prognosis and follow-up for this patient. As a conclusion, based on the scope and finding of this study, we concluded that the artificial intelligence in the form of virtual reality group showed a significant improvement effect in unilateral spatial neglect compared to the control group. Thank you for everyone, and I'm ready for any questions.
Yes, thank you, Dr. Lama. Any questions from any audience? Okay, no questions. Thank you, doctor.
Thanks, doctor.
The next presentation is Dr. Milan Fiala. He's having a technical issue and is trying to log in now. So could you please bear with us for at least 2 to 5 minutes? Dr. King Jong Kong will join in 5 minutes. Please bear with us. Thank you. Hi, is Dr. Andrea? Dr. Andrea, can you hear me? Dr. Andrea, can you hear me?
Yes, I can hear you.
Yeah.
Can you hear me?
Yes. Can you start sharing your presentation?
All right. So share screen now?
Yes.
All right. All right.
Yeah.
Okay, can you see
Stage
the
is yours.
screen?
Can you decrease the percentage of the presentation slide because it's not visible completely?
So how do I do that?
Can you press escape?
Escape. Okay.
Still, the presentation is not visible completely. Yes.
Okay. Can you see it now?
Can go ahead. Yeah. Can you go ahead with this like this?
Okay.
Can you increase this presentation right side?
What's that?
The bottom right side, you can have a drag bar to increase the percentage of the view.
The bottom right side you can't see?
Top right side, bottom right side, not the left side. Percentage you can increase. Right side, please. Right side footer.
Like this?
No, not that one, not that one. This one. You can see the notes. Right side, there is a percentage.
I don't know where notes here. Okay.
Yes. Can you increase it?
Increase the notes.
Yeah. Not notes, not the presentation only I'm asking about. And can you press on this sidebar, minimize this options, file, home, and everything on the top, so that your presentation will go bigger. Right side, yes. Yeah, that's fine. Then you can start presenting now.
Okay, thank you.
The stage is yours. Yeah.
All right, so you ready for me? Can you hear me?
Yes, I can hear you. You can start.
Okay. Well, thank you very much for the opportunity to talk today about GB Science's plant-inspired Parkinson's drugs. I'd like to begin with a little bit of a corporate overview, because at GB Sciences, what we do is what we do and who we are very closely aligned. So GB Sciences is a plant-inspired biotech developing prescription drugs. In our drug development pipeline, we have 5 preclinical programs. For 4 of these, we have obtained animal proof of concept. For 3 of these, the patents have been issued in the US, and they're now beginning to, we have had our first patent issued in China for Parkinson's disease as well. And we have a vibrant discovery program with an intellectual property portfolio that covers greater than 60 serious health conditions. We've obtained overall 6 US and 4 foreign patents that have been issued. And in addition, we have 17 US and 51 foreign patents that are pending. And we also have a proprietary drug discovery platform, which is called Pharos, which combines natural products research of from traditional medical systems, as well as AI and machine learning. So for today's talk, the identification of minimum essential therapeutic mixtures from cannabis plant extracts by screening in cell and animal models of Parkinson's disease. The work that I'm presenting today is also in a publication of the same name. So as I alluded to, GB Science's drug discovery process involves several different components. The first of which is our drug discovery platform, which combines data analytics and machine learning tools. And then we have, we use high throughput screening systems in both cell and animal models of disease. And what we're aiming to do is arrive at plant-inspired minimum essential mixtures. In this case, we're talking about a synthetic cannabinoid API and IP that is for both the composition and the use of these materials. We believe that plant-inspired medicines represent the best of both worlds, in that whole plant medicines have thousands of years of use in traditional medical systems. They usually involve multiple active ingredients that are multiple low affinity ligands, and often more than one disease-related target. However, for whole plant medicines, the plant sourcing is often varied. It may be difficult to achieve strict CGMP manufacturing standards, and these, at least in the US, are often in the nutraceutical market without US FDA pre-market safety and efficacy data. In contrast, the single active ingredient drugs that most people are familiar with are very US FDA friendly. They have a single active ingredient, which is usually a one high affinity ligand and one disease specific target. And the upside of these, of course, is that you have API manufacturing that's consistent, manufactured to strict CGMP standards, and we have a US FDA approval process. What we're trying to do is combine the best of both these worlds. We're trying to tap into the thousands of years of efficacy in traditional medical systems through whole plant medicines, but with the ease of use for single active ingredients by creating minimum essential mixtures, which are a reduction of the numbers of compounds that you'd find in like whole plant medicines down to like 3 to 5. So for our Parkinson's disease, specifically for our Parkinson's disease minimum essential mixture, so MEM stands for minimum essential mixture. We also believe that having multiple components in there is very useful, because when you're trying to address a multifactorial disorder like Parkinson's disease, having the ability to target multiple components of a complex disease can be potentially very effective. As we all know, at the cell level, Parkinson's disease has multiple factors that influence its progression. And these are the widely accepted causes for Parkinson's disease: mitochondrial dysfunction, alpha-synuclein protein misfolding and Lewy body formation, and lysosomal dysfunction. For the compounds that we're using in our cannabinoid-based Parkinson's disease formulation, we have evidence that suggests that they are acting at the level of mitochondrial dysfunction. They're antioxidants, and they've been known to regulate the calcium channels and help with the kinds of pathology that are common in setting up Parkinson's disease, and as well as other neurodegenerative disorders. At the systems level, these compounds in our cannabinoid-based Parkinson's formulations also, we have evidence that they're influencing neuroinflammation pathways. And you're using an assay that's also allows us to track the death of dopamine producing neurons. And we are measuring their influence on the motor symptoms. So big picture, if you look at our drug discovery funnel, for this project, we originally started off with the metabolomic profiles of greater than 1,000, that were greater than 100,000 potential mixtures. And we did an in silico analysis of the cannabis profiles and literature reviews used to select the compounds for studying our complex mixtures. So when we had gone through these potentials, we picked the ones that we thought were the most likely based on our in silico analysis. And then in the complex mixture level, we had around 1,000 potential mixtures, and we used an MPTP screen to look for the best complex mixture. From there, we wanted to the best mixture had 9 ingredients. So we wanted to bring that down to what we're calling a minimum essential mixture. And these are mixtures that contain 3 ingredients that are working together. We screened right around 100 or just under 100 mixtures using the OHDA assay in a zebrafish model. And now we have drug candidates which are in rodent model. So as I mentioned before, this work is reported in this particular paper. And we're going to be looking at both the mitochondrial effects in MPTP assay as well as dopamine secretion in dopamine secretion assays. And so the initial complex mixture results, as I mentioned before, we were looking at in silico screening of cannabis metabolomic profiles. We had 11 cannabinoids and 19 terpenes measured in each chemovar and desired profiles based on in silico data and analytics and literature review. And we then did the complex mixture screening and dopamine and DA neurons. We tested over 1,000 complex mixtures that contain 9 to 12 compounds each, and we rank ordered the complex mixtures based on their cell assay performance. And we chose the best performing complex mixture for further study. We then, as I'm going to show you in a minute, determined that that mixture 1 was a complex mixture that needed to be reduced down to form the minimum essential mixture. In the original mixture 1, there are 5 cannabinoids and 5 terpenes. And so if you're looking at the complex mixture level, one of the striking things that we noted early on was that the complex mixtures seem to be more efficacious than the individual ingredients that they were built on. And what was surprising, though, is that it wasn't just that the major cannabinoids that people were looking at caused these mixtures to be more efficacious. Even without a major cannabinoid, these complex mixtures seem to be quite effective. In we rank ordered them, and in our efforts, we went and did a deeper dive into this mixture 1. And what we found is that if we looked at mixture 1 and we divided it up, mixture 1 contained these ingredients, 3 minor cannabinoids and 5 terpenes. And we looked at the components. We looked at the efficacy of the terpenes separate from the cannabinoids. What we found is that the cannabinoid contribution to the mix seemed to be greater than that of the terpenes. So CBD and CBN were the major cannabinoids that we were looking at. CBC, CBG, and CBDV were the minor cannabinoids, and then the terpenes in this mixture. And what we found when we did the that the mix, each of these with and without the cannabinoids, is that the cannabinoids had a greater contribution than the terpenes. So moving on into the zebrafish, we started to look for the efficacy of these potential minimum essential mixtures on motor symptoms using a 6-hydroxydopamine assay. There's two components to this 6-hydroxydopamine assay. The standard measurement is looking for total distance travel because as the dopamine producing neurons are being selectively killed through the 6-OHDA method, the animals slow down. In addition, we also were able to describe a novel phenotype where there was a tremor-like behavior. I'll talk more about that. So in the move-in studies, as I mentioned, a standard assay uses a center point analysis of the movement of the zebrafish, and it's looking for the total distance that it's moved. However, we were using a percent pixel change measurement in which we were able to look at the quality of movement, and we were able to define periods of basically on and off activity. So when it was in a tremor versus when it was not in a tremor. And so this slide just sets up as I mentioned before, the effect of increasing levels of OHDA on the distance traveled in the zebrafish. And then there's a light dark startle that demonstrates that the animals were responding to a startle in a normal behavior, even though the total movement, when they're not in a startle phase, has been slowing down. And so this is represented in a bar graph way, as well as if you look at the total distance traveled over 90 minutes relative to the control, there was a slowing of the movement. And then, as I mentioned before, we also were interested in trying to quantify the twitch-like activity, which we're calling, which we measured actually based on activities, both activity state switching, so moving in and out of the on and off periods, and the cumulative duration in the activity states. And we were able to demonstrate, once again, that there was a dose-dependent effect on these measurements. So we started using 3 equimolar, looking at the different equimolar combinations. So if you take the cannabinoids that were in the original mix 1, and you just randomly assort them into mixtures, so the first, second, and third cannabinoid for each, and then tested those in the zebrafish model, what we found is that 3 of the possible MEM reduced Parkinson's-like symptoms. And so we moved forward with that, and we also demonstrated that it also, not only was there a reduction in the total distance move as in the previous slide, but also it also reduced the tremor-like activity, and it was concentration dependent. And then we looked to find an optimized ratio. And so we tested, so the way you describe to look for these is you're looking at the first concentration. So CBD would be here. The CBDV is also here, and then the CBC is here. So that's how you describe these. So we looked and we found that 22 cannabinoid ratios were able to positively influence the reduction of Parkinson's-like symptoms, and 5 of them actually outperformed the equimolar MEM. So we moved forward with the optimized ones, and so this is a representation of the optimal mixtures. One of them is an equimolar mixture. So we've selected the highest performers of these 6, and these are now in rodent studies. So in conclusion, there was significant Parkinson's motor symptom reduction in the OHDA model. We were able to demonstrate that the MEM restored overall movement levels and reduced resting tremor, but maintained a normal startle response. And there was a statistically significant reduction in Parkinson's-like motor symptoms. 3 of the 10 equimolar, 22 of the 63 cannabinoid ratio, 5 outperformed the other MEM, and 3 have been selected for formulation and adment testing. So what are the next steps? So we've just recently completed a dose response study for the most effective MEM in rodents at the University of Lethbridge. That data will be available to us next month. And we've formulated clinical prototypes of the most effective MEM in the Zydis tablets that are made by Catalent Pharma. In addition, we're preparing to run these most effective MEM in toxicology and ADME studies in preparation for a pre-IND and IND to get into first-in-human clinical trials. And we really believe that the Zydis oral disintegrating tablets will be an excellent delivery method for Parkinson's patients because greater than 50% of them have problems with swallowing. And there are potential clinical advantages to using this delivery format. So in summary, our plant-inspired minimum essential mixtures are produced by reducing the number of compounds that you would find in a whole plant mixture. And in this particular case, it's using the cannabis mixtures. So sequential screening preserves molecular synergy in the plant's extracts. And we've got it down to 3 active pharmaceutical ingredients in each minimum essential mixture, which can be manufactured under CGMP using synthetic API, which are homologs of the plant compounds. We have done work on the mechanisms of action and a more complete model of biological targets regulating a biological disorder can be performed using AI and machine learning tools. This is not a one drug, one target approach. It's more complicated, but it's not unknowable. And we're utilizing proprietary oral delivery methods for increased bioavailability and increased patient compliance. We have excellent research and development partners who really are bringing this project forward. We have some references, and other than that, thank you very much for your attention today to this talk.
Hello, is it time for question and answer, right? Hello?
Yes, hello, Dr. Like. Any questions?
Can you hear me? Can you hear me?
Yes, yes.
Yes, yeah, this is Kong from Case Western Reserve in the United States. It's an excellent talk, outstanding talk. I'm actually very impressed by the type of work you do. It's a very difficult kind of research. So just maybe I missed it. So all the minimum essential mixture, all the components in the mixture are identified, right? It's not just a not totally identified mixture.
Exactly. So we are using totally identified materials. Yeah. So unlike in plant extracts where there may be ingredients that are just in there, we have taken and used a natural products approach in the discovery side, but when we produce the compounds for the clinic, we're using synthetically produced homologs of the original plant ingredients, and we're putting them in there at specific ratios that have been determined by our studies to be the most effective.
Wow, outstanding. Otherwise, you said you're trying to deliver orally to the mice for the study to study effectiveness, other aspects. So the oral delivery, how do you control the mom? Because orally it's hard to control how much the animal takes.
The tablets. So we did a rodent study. So the rodent study actually that just finished was using a hydrostolic pump right into the brain. So that data is, I was hoping that data was going to be ready for this talk, which it was supposed to have been. It's confirmed that they're finished it, but they haven't got me the final report yet. So that one isn't delivered orally. What we are doing though is we're setting up a study where they're using mini pigs to do PK and PD studies. And so in the mini pig, the oral dissolving tablets work much better.
I see. So mice is delivered directly to the brain.
Yep.
Okay. All right. That's very impressive. So okay, I look forward to hear outcome of the data.
Me too. They were supposed to have gotten me the report 2 weeks ago.
There are people who do this kind of level study mixture of impact on a particular disease, especially disease like Parkinson's disease. Actually, I'm also studying Parkinson's to some degree, but I'm looking at the diagnostic side, not the therapeutic side. Thank you. Great talk.
Thank you.
Yes. Thank you, Dr. Andrea.
Yes.
Is there any other questions? If not, we can go ahead to the next presentation. Dr. Kim Jong, now it's your turn, and can you start sharing your screen?
Just a second. Let me open up my files. Slideshow. Okay, just a second. Screen. Here. All right. Can you see it?
Yes, put them in presentation mode.
Let me see.
Press F5.
Press what?
F5.
F5.
F5 button. There is a key in your.
Yeah. Let me see. No. Control F5. It did not seem to do anything.
Yeah, and in the footer right hand side there is a drag bar of percentage view. There is a funnel kind of picture. Click on that. In the bottom right side. On your screen,
Right side?
bottom right side on the presentations. On the PowerPoint slides. Yes, beside that button. The funnel kind of button. Funnel. The right one. Right side to that. Yeah, right side. No, in the right footer. In the footer.
Right side.
Right side footer. Down. The funnel kind of picture. Right side.
Okay.
Before minus.
Before minus. Oh, here.
Yeah.
Oh my gosh. Okay. Thank you.
Yes. Yes. You can go ahead now. The stage is yours now. Thank you.
Okay. All right. Sorry about the other trouble. It's I wish I could be there in person. But today I'm gonna. This is my name is Kong. Kong. I'm from Western Reserve. Just mentioned earlier. I'm going to talk to you about our study developing effective gene therapy for first of all Jacob disease (CGD), which is the prion disease in humans. So I'm going to first give you some introduction, probably more detailed one, because you guys probably most are not in the field about the prion protein itself, the prion pathology and the prion disease in human animals. Then talk about a little bit on the past efforts for other lab developing therapeutics for prion disease. And then mostly focus on our efforts to develop a effective gene therapy for this type of disease in a humanized mouse model. Prion disease, also named transmissible spongiform encephalopathy or TSCs, are a family of very diverse, but always fatal and usually transmissible neurodegenerative disease that affects humans and many several mammalian species. The clinical manifestations of prion disease include behavioral changes, cognitive deficits, including memory loss and dementia in the later stage, and also include a disturbance in motor functions like called ataxia, oscillating gait. And then also some people have the speech difficulties and vision issues. In the brain, the pathology usually shows spongiosis. That means speculation or loss of neuron other cells there. That's kind of, if you look at the slides, you can see neuronal loss. Also gliosis, most of the actual gliosis. And also the one hallmark of the prion is you're going to see the prion deposits or plaques, basically the aggregated prion protein in the brain. And the one hallmark of prion is also that the incubation time, if he acquired it by infection, it can be very long, can be up to 60 years after exposure. So this can be something very hard to monitor, and sometimes identify this original source of the infection. Prion is a term coined by Stanley Fursener, who discovered this disease group. It means this is the proteinaceous infectious particle. which means it's infectious particle that's made of RNA proteins. It can also be indicated as a PRP, which is prion protein, as semi-scrapy, which is what's hallmark of one of the prototype of prion disease. The PRBSC, which is actually the PRP protein, is the causal agent of prion disease. That has been confirmed again by a ton of studies. Prion, up to today, is still the only non-transmissible agent with no required specific nucleic acids. Although some other disease, other disease like the A-beta, tau, or alpha-synuclein aggregates, also the SOD1 in the LS, also show prion-like properties, but they are not considered transmissible, or that is not infectious between individuals. So they're called prionoids. Only the prion disease are real prion that are transmissible. There's no treatment for any prion disease, human, animal, so they're all fatal. That's one reason we want to develop some therapies to help patients. This is a kind of brief summary of the major types of prion we know of in human animals. In humans, you can see there are many types because it's studied most. The most common type is called Creutzfeldt-Jakob disease, or CJD. Among the CJD presentation, it can be divided classified into sporadic form, means we don't know where it comes from. Then there's familial form, basically genetic. And then the third form is acquired, it's from outsourcing fashion. This includes so-called iatrogenic CJD, which is mostly coming from like in the early time in the 1960s, 70s, there are people in Japan and Europe, especially France, they in order to increase the height, they inject growth hormone isolated from cadavers, from human pituitary cadaver tissues. And some tissue apparently contaminated with CJD prions, and then the patient received the injection of the hormone isolated that way, becoming infected. Then the second type of acquired CJD are called variant CJD, which is infected through consumption of macaque beef. That's probably most people know about this. And the last one major type is called Kuru, which was due to cannibalism in a primitive tribe in Papua New Guinea, where they practice eating their dead relatives. And that some relative had a CJD, so their who consume it can become infected. That's where we know from that study that the incubation time can be as long as 60 years from time conclusion. And then the second class of human prion is called Guzman-Strauss-Schinkel disease, or GSS. This type of prion disease are all due to genetic mutation in our prion protein gene. The second, and the other one is fetal insomnia. Some patients are from genetic mutation, some others could be sporadic, but they have similar presentation. And the last group is variable protein-sensitive prionopathy, which is I'm involved in the discovery that subclass, which is unique in a sense that it's localized sporadic, but it has a unique presentation that their prions look different. So in animals, the cattle, we had mentioned earlier, is macaque disease or bone spotting adenopathy, which is well known because it's causing variant CJD. Although today, most died down already. The current problem with animal prion disease is actually the chronic wasting disease in the cervid population, which include deer, elk, moose, and reindeer. This type of disease in this animal are a very big problem in the United States and Canada. More than half the states in US have this problem in their wild and or farmed cervid populations. And in Canada, there are 3 provinces have this problem. In some area, very, very severe. Whether this prion transmitted to humans is not totally sure. I'm studying that. We have some other suggestion maybe, but there's no definitive evidence yet because we have not found a patient yet. Then the prototype of prion disease are scrapie, which is the prion disease in sheep and goat. This type of disease has been recorded in literature, in the history, in history books that at least around for 1,000, 2,000 years. And that's because the animal have the symptom, because they are symptom itchy, they cannot scratching, rubbing against some rocks or trees. That kind of sign is quite scrapie in some sense. And that's very common. Most countries have it. And a few countries like New Zealand don't have this problem in their sheep goat population. And the last major type is the transmissible mink encephalopathy, which is present in the mink. This is most severe in the U.S. Something I think last 1960 something that they had problem in their a huge problem with this animal, and some people believe this mink prion maybe is the source of the CW chronic disease prion, but there's no definite evidence at this point. So I mentioned earlier the genetics of prion disease, actually prion disease in general, are due to if it's genetic, it's caused by mutation in the prion protein gene or PRNP gene. This is only known major gene involved prion disease. Although some modifier genes identified, but they play a very limited role in the pathogenesis. In human prion disease patients, the genetic form counts for about 10% of the cases. 90% or more are sporadic, with only less 1% are acquired form. And then tunnel mutations identified prion protein gene that associated genetic disease, but many of them are some of them have high penetrance, some don't. So it's highly variable. They year code disease between 40 to 60 years, whether it's sporadic or a genetic form. They are acquired. I mean, acquired can be early, younger, but mostly between 40 to 60 year age of starting. Here is kind of a summary diagram of most of the identified genetic mutation, which is on top of the line. Those in red are the genetic mutation associated genetic form prion disease. At the bottom, there are some so-called polymorphism. A few of them, like 129 methionine binding, a couple others, that has a big role in modifying the disease presentation or sometimes modifies susceptibility to exogenous prion infection. So the prion protein itself is fairly small. It's like 253 amino acid total in human prion protein. It has second peptide. It's directed to the cell membrane, and it has an N-terminal off-target repeater gene, which is important for genetic disease if it has a change in the number of repeats, which kind of many disease like Parkinson's or Huntington's, particularly many other disease that are caused by mutation expansion oftentimes. Here is a similar case. In this case, the off-target region can be expanded, cause genetic prion disease, and those other permutations are common. You can notice the C-terminal part. So there's some small short beta sheet region and several or three alpha helical region. This part, C-terminal part, is more structured. It's a globular domain, which, including part of the central part, are essential for the prion replication. And then this other part, there's a GPI anchor, the C-terminal end, that allowed it to attach to cell membrane through the GPI domain or moiety. So here is kind of diagram of the structure or combination of the cellular prion protein. This is this C-terminal part, the GPI part, and then the alpha part, and then there are two big globes. That's what are the glycans, such as prion protein. The prion protein is they mentioned there is a central player in the prion disease. It's essential not only for prion replication as a substrate, but also essential prion pathogenesis. It serves as a receptor for toxic signaling for prion disease. So it's definitely absolutely essential for prion disease. It's a highly conserved protein, and it's almost ubiquitously expressed in all cell types. It's just expression level is different, mostly concentrated expression in the brain, muscle, and lymph tissues. There are many functions assigned or implied to be conducted by the cellular prion protein, and this is some important role, including cell transduction, neuroprotection, and maintenance of the peripheral axon mining sheaths, as also involved in cell differentiation, like immune cells and other cell types, and even in differentiation for muscle cells. So it has many roles. However, the prion protein is not essential. If you knock out the prion protein gene, the animal, or like we have knockout mice, also they're knockout cattle, they appear large and normal, so it's not essential. This is why it makes it a very attractive target for manipulation for therapeutic development. It's safe to play with. Another important factor about serum protein PRP is that it plays also a critical role in cancer, oftentimes promoting cancer, and also in several more common neurodegenerative disease, like Parkinson, Alzheimer's disease, Parkinson's disease, Parkinson dementia, and ALS. The role the PRP plays there mostly is that it plays as a receptor. Again, as a receptor to mediate the toxicity carried by these so-called oligomers of those toxic protein involved, like in case Alzheimer's disease, A-beta, in the tau, and then in case Parkinson's, alpha-synuclein. So the PRP play a very important role in many diseases. That's why we're targeting it. We're hoping that we develop a strategy that can target all disease, not just Parkinson's disease. All right, I just said that penalties, the PRP is the central player, the pathogenic agent is the PRPRC, which is basically misfolded aggregates of PRP protein. And the PRPRC, in fact, animal humans also concentrated in the nerve tissues and in lymph tissues, a little bit in the muscle, or maybe occasionally in the heart. This aggregate is very stable, means it's very hard to be disinfected. The reason is because when it converts from cellular form to the aggregate form, of course, it becomes aggregates, and it becomes insoluble. And if you do protein digestion, normally the protein is show up on visible as 3-band because it has 2 glycans. It can be both glycans inside be attached by glycans, or just one side has glycans, or there's no glycan. So generally speaking, you have when you do splice, look like 3 bands, depending on the glycosylating status. And if you digest with the proteinase or protease, the flexible end-terminal part protein will be degraded, and you leave the protein resistant core part. So there's 3 bands, but they are smaller. And the size and the pattern of the 3 bands are also a very important feature to identify or distinguish different prion strands sometimes. Now just that I'll talk about the prion strand later. So how does the prion replicate? We know that a normal cellular property is substrate for replication. So this is a model called seeded nucleation. Basically says, although most times cellular property adopt the normal configuration, but they're very small propensity for some molecule will adopt the disease conformation. And the equilibrium oftentimes usually is kind of severely tilted towards the cellular form. But when something happens, for example, when there's mutation in the prion protein gene that make it favor more towards the disease conformation, the band will shift over there. And then the disease form tend to aggregates, the conformation 10 aggregates. And then when it reaches a certain size, becomes stabilized, called the seeds. The seeds will pull the equilibrium towards the aggregation direction, and from larger aggregates. This large aggregate will be break down process to produce smaller fragments, and these smaller fragments serve as more seeds to amplify the replication process. That's how we believe prion replicates. As I said, the prion protein misfold from aggregates, but the form of misfolding, you have different ways to misfold. So they can have a different conformation of the aggregates. That's what we believe is the basis for prion strands. I'm not going to do detail about it. The prion strands actually are not associated with different conformation. They also often associated with distinct pathology and sometimes distinct disease as well. That's all the prion diseases in different animals, in humans, they all have different strengths, different molecule strengths, especially humans. I don't know, right now we have at least 8, 9, 10 different strands already. In animals, also at least 3 strands in different species. And this presents a challenge because the different strands, as I say, have different pathology. They may respond differently to different molecules. If you develop therapies, it may be whatever molecule you develop maybe is effective for only one strand, but not for others. This makes it very hard to apply clinically. And the one feature of other disease like Alzheimer's, Parkinson, their aggregates also have different conformations, and there are recent studies show that it also have different strands that's associated with distinct disease or pathology. So they have a lot of similarities there. very recently, actually a few months ago, that the prion conformation actually was resolved for a couple prion strands from rodents. You can see that they actually adopt so-called parallel in register beta beta sheets, meaning the whole protein, the whole amino acid chains are formed beta sheet conformation. But so the other one monomer after they stack on top of each other in the same direction from this kind of structure. But how does it encode the strain information? The thing is, you see there are two sides from the cut in the middle. The two sides, there's kind of one single chain region in the middle here, and they can kind of has a flexibility, it can fold like here. This one has more open up, the left side strand more open up. The left side, the right side, the RMR strand, they kind of fold up more. So this kind of open up or folding of the kind of two halves are, we believe, are the major driver of different conformation or different strands. So this have a huge implications there of how we identify how animal, how prions infect different hosts or spread within hosts, and how we may develop drugs against it. Actually, their study already tried to design vaccines based on structure to see whether they can control it, with some success. So let's get back to our main focus is to try our effort to develop a therapy, because we said this disease is universally fatal. Previous efforts are mostly focusing on the developing small molecules, like many other effort development drugs against other disease. Mostly those are anti-prion compounds, so-called. The famous one include the quinaprine, doxycycline, and penicillin polysulfate. This molecule, generally speaking, they kind of inhibit purification, or they destabilize the prion aggregates. And these molecules, they all show very good effect in rodent models against the rodent prions. But unfortunately, they all failed in human clinical trials. Basically, they show no effect at all, kind of in human patients. So it's showing difference between different prions, between different strains. That's kind of very disappointing. So the another effort is trying to develop antibodies against the prion protein or against the prion, pathogenic prions. They actually, the recent clinical trial conducted by the Collins group in the UK, and they a few patients actually. The patient very rare. There are a few patients there, but results is inconclusive. And the worst problem is that it did not show clear clinical benefit yet. And the other concern is they use one particular type of humanized antibody. They seem to don't have much toxicity, but many other anti-prion protein antibody actually are very toxic. So when you use this strategy, be very careful about how to use it. And then the third approach that seemed to have promising results, at least in mouse models, is called antisense oligonucleotide approach. Basically, designs a short oligonucleotides that target the prion protein mRNAs. This is going to use general strategy called RNA interference strategy. Basically, you use some small piece of DNA or RNA molecule that can bind to the sequence of a particular mRNA that promotes the degradation, so that can knock down the. So this approach has been tested in two recent studies, mostly actually from one lab in at Harvard and MIT, and at Harvard, the Broad Institute, the kind of joint thing there. They show that it's pretty effective against the 5 different strains, mouse strains though, in rodent models. So that looks promising. However, nobody has done an ASO study targeting human prion disease in human prion disease models. And this approach, even if it works, it will require repeated high dose ASO injection by injecting directly in the brain or to spinal cord, in order to be effective. So this kind of repeated injection, that kind of sensitive side, I think also present serious safety concerns if it works. So we are trying to develop a service strategy that targeting prion protein as well, but we hope that it will be safer, more effective, and also broadly applicable to not only prion disease, but also to Alzheimer's disease and Parkinson's disease and a few other diseases that where the prion protein play an important role. So the first strategy we're adopting is knocking down the cell surface prion protein in the central nervous system, which is similar to the ASO approaches that we use in gene therapy. The reason I said it because the brain prion protein is essential for both prion replication and prion pathogenesis, and it is dispensable, so we can play with it safely. And I mentioned earlier, an ASO approach already shows it works at least in mouse models against the rodent prions. But we are focusing on developing a gene therapy approach using either hRNA or siRNA against the human prion protein. This siRNA is similar to SO, but it just expressed from a gene, and it produces small piece of RNA that can, or similarly like SO, can target the PRP mRNA for its degradation to knock down PRP gene expression that way. And the second approach is you instead of degrade mRNA, we try to enhance the cleavage break the protein into pieces or just the release from cell surface. That's our approach. So before I go that, we have to talk about the cleavage prion protein, which is it plays a lot of important role even normally in physiological cases and pathological situations. So this for cellular prion protein PRBC. And we mentioned earlier, this is the diagram of this protein, and it's essential for purification as substrate. It's also essential as a receptor for oligomers of prion aging, PRBC, and also important receptor for A beta, tau, and alpha-synuclein. It could be potential for other areas of other disease as well. And so, because of this role of prion cellular prion protein, if you knock it out or reduce to the level, you reduce the substrate, so you can inhibit replication that way. You also reduce toxicity because the receptor is also reduced. So you kind of double whammy against the disease. And if you do cleavage, there are three major cleavage: alpha, beta, and shedding. We can focus on alpha cleavage because it cuts right in the middle of the protein, produce two fragments, N-terminal N1 and C-terminal C1 fragments. These fragments are also beneficial because the C-terminal fragment C1 can inhibit purification. The N-terminal N1 fragment is neuroprotective. Actually, it's not just neuroprotective; it's protective against other stressor as well, and it can also directly neutralize toxic oligomers like A beta oligomers. And this cleavage, because of cleavage, the two fragments N1 C1, they could no longer serve as substrate for purification. So this alpha cleavage is also believed to be highly beneficial because it does all the things. It reduces substrates, it produces two beneficial fragments. So there's nothing bad, all good, looks like. And I'm going to show you later that we identify, but the problem with people don't know what enzyme does this alpha cleavage. We identify in my lab, we believe that ADAM-A is one major enzyme that does alpha cleavage. So that will help us to develop a strategy for therapy utilizing alpha cleavage for the treatment purpose. So how do we study the therapeutic effect? So we have to have a mouse model for that. So here we developed a humanized mouse model for CGD, which is very simple. We're just using a mouse genomic clone that containing the mouse PRP gene. We just replace the mouse PRP overloading frame with the human PRP overloading frame. And then we knock out the mouse gene, just leave the human PRP gene expression. That's how we produce our humanized mouse model. The reason we can do this is because we know that for prion disease, the overwhelming deciding factor is the prion protein gene itself. Other things doesn't matter that much. So by just the single gene replacement, we can make a pretty good model for human prion disease in the mice. We show that actually. So we make this transgenic mice, we call TG1. We inoculate with the sport CGD, the prion, or MM1, that's the one, but the most common one I did not mention earlier. It's the most common prion in humans. And we see that in the mice, we do West blot, look at the prion combination in the brain. You see the prion in the mouse brain is basically exactly the same as the one you put in from human patients. And then you can quantify the different glycoforms, and you can see they're basically exactly the same. Besides, you can look at the pathology, as we talk about the spongiosis, but the holes in the brain, in the cerebral cortex, is the one like you see in the human patients. And the cerebellum is no damage. They're all cells, neuron cells are there, like a Purkinje cells and molecular granular cells are all there. They do not touch the cerebellum for this particular type of spongiosis. And with relatively short prion equine time for prion disease, it's like 60 months. It's considered short. Unfortunately, this type of study need to take a long time. Costs a lot too. And if we look at the prion deposits, you can see this is kind of those dark brownish dots or kind of granules. Those are the prion deposits. And this kind of fine, we call it synaptic type of prion deposits, are a signature of this type of spongiosis in human patients as well. So we believe our model faithfully reproduce the pathology and the prions in the mice model. So it should be a very good model to study human prion disease, especially for the M1 subtype. So now we talk about our approach to develop therapies. Here we're using siRNA approach targeting human PRP mRNA, with delivered with recombinant adeno virus. You probably know that there are several gene therapy has been approved for treating human disease, single gene mutation disease, human patients, and they all except one are delivered by the recombinant virus. Is because this vector are very safe, and also its effect lasts for years after one shot, maybe longer. Sometimes it may be decades. So this is why people use this RNA vector to deliver. We adopt this same vector for our study. So we designed several siRNA, that means we short helping RNA against human PRP mRNA, and we done 4 of them, put in different vectors. We test their effectiveness first in human neuroblastoma cell line called M17. You can see that this is normal prion protein without any treatment in the cell line. And if you scramble the siRNA, you have little effect. But with these 2 targeted siRNA, 1 and 2, it knocked down PRP a lot. And we quantify here on the right side, you see the knockdown PRP level by around 90%. For all 4, I'm not sure 2 here, but all 4 have around 90% knockdown. It's hugely effective. And that's what we use plasmid in cell lines. But in vivo delivery of plasmid is not effective. We have to use RNA as vector. So we adopt a RNA with capsid B10. But this is a artificially derived capsid that is very effective in crossing blood-brain barrier. So we just need to inject the RNA virus prep containing this RNA sequence, rich orbitally. Basically, it's like an intravenous injection, basically into the blood directly. And we show that after this kind of one injection, we look at the level of brain PRP in those mice without of this injection. We can see about reduction of PRP level by around 26% after giving them around 10 to 12 viral genome copy. That's a dose. So it seems that the knockdown is not that effective, only knockdown by 26%. But we can show later it already has scientific effect. As is shown here, this is mice we use T40H mice, which is a humanized mouse and overexpressing, means expressing more than normal level of PRP, human PRP in the brain of mice. We inoculate with a M1 prion from human patient. Then with treatment, we can see that the survival time is extended by around 10%. But the thing is, this is probably the best we've reported so far. Nobody has achieved this level of protection when treatment is given. Here is almost is just before, or actually some mice already have early clinical signs. So already clinical sign already occurred, or already almost there. We show this effectiveness. Is the first thing of this kind. So we are very hopeful that it will work. I'm giving more reason why we think it's more hopeful than it is shown here, because we use only one type of capsid, which is the B10 capsid to deliver it, and this B10 capsid is the first time used in this particular strain, this mouse strain background. There's another capsid called PHP-EB, we call EB capsid here, RAE capsid here. And then we use this capsid here to deliver this SRN2 or SRN1 in the same T4D mouse brain. We achieved, for SRN2, we achieved over 52%, almost 53% knockdown of the PRP. Another SRN1 is a little bit less effective. So if we just change a more effective capsid, we achieve much higher knockdown effect. More importantly, or equally importantly, if we use a different mouse strain where the capsid is more effective, even still the P10 capsid, still the same RA, IRV, SRN2, we use only 1 to 10 to 11th viral genome copy, which is less than 1/10 of the one used earlier with the in the T4S mouse strain. We introduced in the black 6 mice, which is the one type, the commonly used wild type mouse strain for many studies, for cancer immunology studies. So with this, we achieved, just by this small dose, one injection, we achieved 57% knockdown of PRP already. So you imagine if you give it 10 times more viral genome copy, it's going to have even higher knockdown effect. So if we achieve it, so that gives us a hope. If we give this RAV siRNA with the proper capsid in human patients where the PRP level is normal, not overexpressed, we should achieve like 60% or more knockdown of PRP, which means they're going to have the effect of extending the life survival by at least 100% or more. So we are highly hopeful our approach will be actually useful when we come to the clinical trial stage. So that's one approach. The second approach you mentioned is instead of knockdown PRP by siRNA, we can try to enhance our cleavage of prion protein. So this is where I show here. I told you earlier that ADMA, this enzyme can cleave the PRP at the alkylated site to produce two beneficial fragments and then reduce PRP substrate at the same time. So this is what I show here. This is the same guy in the M7 cell line. We introduce plasmid expressing ADMA. You can see that we look at the alkylated activity, which we show as the ratio between the 4 nanometer PRP, which is on top, versus the cleaved C-terminal fragment C1. The ratio is a reflection of the alkylated activity. We show here, when you express ADMA, the alkylated activity shot up by 4 or 5-fold. So it's a huge effective in terms of enhanced cleavage with this enzyme. And in a more relevant model where in the primary neuron isolated from either wild type mice or where the mice with the ADMA gene knocked out, you can see that if you knock out ADMA gene, the alkylated activity dropped by 60%. So it's play a huge role in the neuron, at least, that the ADMA play a huge role in the neuronal PRP protein alkylated. And in mice, if you look at the mouse brain, compared with the brain in the ADMA knockout mouse versus the wild type mouse brain, you can see that in knockout mouse brain, the actual PRP alkylated activity dropped by about 30%. So it's less than the primary neuron, but it still play a major role there. So so that's the how do we that's a second strategy is elevated extracellular level of the actually. So the next step we're going to do is trying to design make a RAV viral vector that express the ADMA gene and introduce in the mouse brain, as we did it for the siRNA, and let's see how it works. So this is in progress. So now let's go to second strategy to evaluating to elevate extracellular level of the neuroprotective PRP forms in the brain. So this is that's our rationale behind this strategy, because the cellular protein, prion protein is a GP anchor membrane protein there, and it goes through our cleavage, we call our secretase, which basically ADMA, and mostly we know of at this point. You produce C1, which is inhibiting proliferation, and then put AN1, the N-terminal fragment, which is a neuroprotective. You can we have actual data to show that AN1 can suppress proliferation directly. It can bind to A beta to suppress the A beta aggregation, and it can also bind to already formed beta aggregates to make it a non-toxic. So it does same thing with like a tau aggregates as well for autopsy or frontal dementia. So it is a very useful molecule produced by enhancing our cleavage. And how can we enhance the accelerate the neuroprotective A1 peptide level? There are two ways to do it. One is enhance allocate, right? Enhance level our secretory activity. In our case, you can enhance ADMA activity level, or it can introduce the A1 peptide directly by injecting, for example, recombinant A1. But unfortunately, A1 peptide is very unstable. It is a very short half-life. So injecting recombinant protein is not viable. So the other approach is you design a gene separate vector, express the A1 peptide from a gene separate vector, so you can make it biofactory in vivo to provide the A1 peptide that way. And that's our approach we're trying to adopt. But unfortunately, we're trying to express A1 peptide directly from a vector. We try first in the muscle cell line, CL12. We cannot produce anything. We cannot detect anything. So the A1 peptide by itself directly does not work. It cannot get out of cells. It never found out that the A1 peptide is kind of got stuck inside in the ER, so it cannot get out. That strategy died. So to address this problem, we make a so-called variant of A1 called A1 plus. That actually deliver a larger form of the A1 N-terminal fragment. We show that by doing that, we can express from a plasmid and express it both in the neuroblastoma M779 and also in the muscle cell line CL12. So this is we have detected in both in the cell body and in the medium. Means this actually got secreted to the medium, which is what we want. You want to make a biofactory, not let them stuck in the cell, but I want to get out of cells so can diffuse throughout the brain to provide protection. We did an injection of the plasmid expressing the A1 plus peptide into the brain, and we showed that actually detect some of the A1 peptide actually in the blood even. So we think we have achieved expression by even using plasmid. So now we are making RIV expressing the A1 plus peptide and hoping that they will achieve high level of axillary level of A1 plus peptide in the brain of the injected mice, and together with the RIV carrying sRNA is going to have even better therapeutic effect. That's what I said. This is therapeutic strategy 3 is combine the two together. All right. It's from what I told earlier, our approach is tightening PRP, but knocking down PRP level, and also provide new protected A1 fragment, or A1 plus in this case. It should apply not only to prion disease, but also apply to other diseases that prion play an important role in pathogenesis, like Alzheimer's and Parkinson's. So, so I'm hoping that we, with the same approach, can target several major diseases at the same time. We are working toward that goal here. So in conclusion, we believe that the brain PRP knockdown by a single systemic RNA delivery that is a very promising gene therapy strategy for clinical surgery treatment. And then second simultaneous knockdown PRP and overexpression of the new protective A1 fragment or N-terminal fragment PRP should be better, offer better treatment options. And I hope that this strategy can work against the other disease like Alzheimer's disease as well. So now thank people in my lab and my collaborators who most help with the N1 the N-terminal fragment project, and the funding agents from NIH and the CGD Foundation. And thank you for your attention. Now I could enjoy some questions.
Yes, thank you, Dr. John. Any questions from audience? If there are no questions, then we can move ahead with the next presentation.
Thank you.
Yes. Dr. Milan, can you hear me now?
Yes. Good morning.
No.
Can you hear me?
Yes. May I know is this? Is this Lara?
Can you see me and hear me?
Yes. 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 May I know is this? This is Dr. Lara.
My name is Laura Milla. Yes.
Yeah. Laura, can you wait for some time? Like there is another presentation is there, Dr. Milan. So after that, you can
Yes.
present.
Can you see the slides?
Yes. There is another presentation is there, Laura. Can you wait for some time?
Yes. Good morning, good afternoon, or good evening. My name is Laura Milla Sanabria. I am a professor and researcher at Universidad Nacional de Río Cuarto, Argentina. I want to thank Scholar Conferences for inviting me for the second time to this prestigious World Congress. Today, I'll be talking about photodynamic therapy for the treatment of glioblastoma. Glioblastoma is the most malignant type of brain cancer. The symptoms or signs of the presence of this type of tumor are increased pressure on the brain, headaches, seizures, memory loss, and behavioral changes. It tends to grow rapidly and spread to other tissues, and conventional treatments for this tumor include surgery, radiation, and chemotherapy. Despite advances in the therapies, the prognosis remains bleak, and patients tend to have a life expectancy of around 14 to 15 months. Glioblastoma treatments are ineffective to eradicate all the tumoral mass due to surgical incapacity to remove all tumor cells, most notably diffusing toward the most notably the infiltrative ones. Moreover, the blood-brain barriers that can prevent drugs from diffusing toward the tumor further complicate the effect of chemotherapeutic drugs to reach these cells. Also, both chemo and radiotherapy cannot easily eliminate resistant cells. Photodynamic therapy, or PDT, consists of the activation of a photosensitizer drug by light of a specific wavelength, which leads to a series of photochemical reactions that allow the destruction of the tumor by oxidative stress. PDT has been investigated as an adjuvant for the treatment of glioblastoma for approximately 40 years, and it has gained attention as an alternative treatment of this disease. Based on clinical trials that report improvements in the therapeutic response, photodynamic therapy has been approved in Japan for high-grade gliomas, and it is expected to be a valuable advance as a more selective tumor cell therapy for the infiltrative tumor area. Drugs used in the treatment of glioblastoma by PDT include the classical photosensitizers phorofrin, laserferin, Foscan, and Levulan. Levulan contains aminolevulinic acid, or ALA. ALA is a pro-photosensitizer, which is taken up by cells and then converted into the photosensitizer protoporphyrin 9 through the heme synthesis pathway. Cancer cells have enzymatic failures of this pathway that favor the accumulation of the intermediate metabolites, mainly protoporphyrin 9, as we can see in the photograph. So the fluorescent property of the photosensitizer is used for photodiagnosis and fluorescent-guided resection of glioblastomas. The surgical resection guided by the photosensitizer's fluorescence prolonged the average survival of patients. The Food and Drug Administration approval of ALA for fluorescent-guided glioblastoma resection renewed interest in leveraging this agent as a means to administer photodynamic therapy. Remaining cells can be irradiated to remove as many cells as possible. First, ALA is administered to the patients. Then, the surgeon removes the tumor aided by photosensitizer fluorescence, and finally, the remaining cells are irradiated to cause their death by oxidative stress. But unfortunately, some cells always resist treatment, generating recurrence in a few months. Studies on glioblastoma resistant to PDT could be of value in improving treatment outcomes. So the objective of our research is to know the characteristics of glioblastoma cells resistant to this therapy in order to plan strategies for their elimination. In our laboratory, we obtain cells resistant to PDT by applying several treatment cycles. Now we are using a glioblastoma cell line called T9HG. We are considering using other glioblastoma cell lines and other models in our studies as well. Our PDT-resistant model consisted of applying 7 rounds of treatment to T9HG glioblastoma cells. The cells that never received PDT were called parental cells, and the cells that received 7 cycles of PDT were called resistant cells. We observed that the resistant populations accumulated less photosensitizer, formed spheroids of higher number of cells, and had higher tumorigenic capacity, and express higher messenger RNA levels of some growth factor receptors than parental cells. We believe that proliferation and tumorigenic molecular weights will be positively selected or induced by PDT. The studies of glioblastoma resistance to PDT will help to better understand the causes of tumor recurrence after treatment and to develop new therapeutic proposals in this field of oncology. At the moment, we are studying the implication of growth factor receptors in the survival and migration of glioblastoma cells to PDT and the possible advantages or disadvantages of using an ALA derivative compared to ALA for fluorescent-guided resection of glioblastoma and for PDT. We are also carrying out some experiments on the response of tumor stem cells to PDT. We want to find out if glioblastoma cells of tumoral stem type confer resistance to PDT. We must not forget that the problem of glioblastoma is complex due to the inability of treatment to remove infiltrative and resistant tumor cells, the influence of the tumor microenvironment, and the presence of the blood-brain barrier must be taken into account. It is our intention to be able to contribute to improving photodynamic therapy to prolong the survival of people suffering from this disease. I want to invite you to read our last 3 publications. One of them is the work on the initial characterization of glioblastoma cells resistant to PDT. The others are 2 bibliographic review publications about the tumor biology and PDT, and about what is known up to now about tumor stem cells and PDT. Finally, I would like to thank CONICET, Universidad Nacional de Río Cuarto, FONCIT, Instituto Nacional del Cáncer, and also Guido Torres, Velázquez Pruca, Nigra, Casas, Urbina, Bellini, and Scholar Conferences. Thank you.
very interesting talk. Maybe I missed it. How do you deliver the light to the patient's tumor in the brain? Do you have to open up the brain so the light can get in?
Excuse me, can you repeat?
Yeah, my question is,
English bad.
since you need to use the light to activate the light sensor agent, how do you deliver the light to the brain, to the tumor?
Yeah. Yes, there are different different mechanisms to irradiate the tumor, employing a ball. The crane is open, and then a diffuser, excuse me, my English is bad. Hay difusores que llegan al tumor con un balón, a balloon, with different compounds that allow light to have a strong intensity. There are small lasers to irradiate the tumor. Excuse me, my English is bad.
You're fine. Okay, so
there are different mechanisms, different irradiated methods.
I see. So you don't need to open up the skull so that your light can shine through. But what if the tumor is deep? Yes, you also need to. I mean, if the tumor is deep inside the brain, you have to maybe open up the brain a little bit also so that light can get to the new tumor. Is that right?
The tumor is
if it's deep inside the brain, not on the surface, how light can get through to the.
No, it is a difficult because tumors generating are deep in the different areas of the brain, and the surgeon have to open the brain and resect the tumor that is deep in the generally are deep in the brain. Then it is difficult to resect the tumor, mainly if it are in areas, important areas. And generally the tumor, the glioblastoma are deep in the brain.
Okay. All right.
It is necessary to develop more efficient irradiation systems.
All right, thank you.
Thank you. I'm sorry, my English is bad.
No, yeah,
Yes,
you're fine. I can understand you. Thank you.
Okay, thank you.
Thank you, Dr. Laura. Okay, so I invite the next participant, Milan Fiala. Dr. Milan?
Yes.
Oh. Thank you for the next. I'm inviting the next presentation.
Hello.
Dr. Milan. Yeah, hi. Can you start sharing your screen, please?
Can you all see me?
Yes. Can you start sharing your screen?
Okay.
And you can also enable your video.
Slides. Can we see my slides?
No. Can you share your screen? Yes.
Oh my God.
So can you double click? Yeah. Can you open your slides? Can you open your slides?
Okay.
Slides.
Sorry.
Can you open your PowerPoint presentation? You can able to see your Outlook.
Okay.
Open your slides.
I am.
Yeah.
Okay. Okay. You see the slides now?
No. Can you open the file? Yeah. Can you press F5? F5 key. F5
Okay,
key.
Alzheimer, this is my presentation.
Yes, can you press F5?
Alzheimer,
F5
what
key.
do I need?
F5.
I'm
F5.
sorry, what do I have to do so you can see it?
Yeah, that's fine then. Go ahead. Thank you.
You can see?
Yes, we can see. You can go ahead.
Okay. Alzheimer's disease clearance by we see that Alzheimer macrophages don't clean the brain as they should, but we can improve the clearance by polyunsaturated omega fatty acids with antioxidants, vitamin D3 to support energy and function. And we also add sialic acid for macrophage receptor glycosylation. So we are treating Alzheimer's disease in patients by improving the immune system of patients. The brain with sporadic Alzheimer's disease is saturated with amyloid beta and phospho-tau because the brain clearance by immune cells, macrophages, and the G lymphatic system fails. And the macrophages lack energy due to the downregulation of energy genes in the OXPHOS pathways. And this happens because of oxidative stress from reactive oxygen species, which are induced when the when the amyloid beta is being picked up. Now, the polyunsaturated fatty acids are something that you all know because they are available and in health food stores, but it is not as simple. The supplementation has to be done 5 days a week, omitting the weekend. And the polyunsaturated fatty acids are signaling molecule. And now I will show you the difference between Alzheimer brain macrophages and control Alzheimer brain macrophages in these two low pictures and the control macrophages. So the healthy macrophages pick up the amyloid beta. As you can see, the green amyloid beta here, they are saturated with it. And they pick it up also on the surface before it's downloaded into inside the macrophages. And these macrophages, instead of having this huge amount of amyloid beta, then concentrated in the lysosomes, and it is degraded. Whereas in the Alzheimer macrophages, the amyloid beta is never downloaded inside. It stays on the surface and nothing is degraded. So what is the problem? How do we handle the problem of Alzheimer's disease therapy when these macrophages are not working right? We have tested macrophages in different concentration of glucose. And we have gone from 100% glucose, which is normal, up to 280%, or we have to hyperglycemic level, or we have gone from normal to hypoglycemic level. And you can see that the phagocytosis, the green cells, is normal in 100% glucose in this subject only, whereas when the glucose, but it is very marginal in all the other concentration. But when we add omega fatty acids, even at 160% or 280% glucose, we're getting some phagocytosis. And we can also show that when the glucose is decreased to down to 10%, and we add here the the omega-3 fatty acids, we are still getting phagocytosis. So we have found that the problem with hyperglycemia, which inhibits clearance of amyloid beta in diabetics, and that's why they are so prone to Alzheimer's disease, it can be overcome by adding omega fatty acids. Now, how does the brain, the Alzheimer brain, look like when it's being cleaned? Well, we have the brain of a patient who died with Alzheimer's disease, and we have then looked for the plaques, which are red. The amyloid beta in all my slides is red, and the macrophages are always green because they are stained by the CD68 marker. So you can see that there is a cleaning going on in this plaque, whereas in this plaque, the cleaning has been already almost done. So there is a heterogeneity, and that's one of the problems, that the clearance is asynchronous, and that there are vestiges of amyloid beta in some plaques. Now, how do the immune cells called macrophages clean the amyloid beta, and how do they get rid of it in the brain? Well, this is a vessel cross-section, post-mortem brain, and the amyloid beta here is in the vessel in red, and then we have this macrophage here that is coming to the vessel and releasing the amyloid beta underneath itself and making the congophilic angiopathy worse. And that is the problem that we want to tackle. But to give you an overview how the brain is cleared in a sketch, here is what we just saw. There is a plaque which is invaded by macrophages that load it, and so they become yellow when they load amyloid beta, and they migrate to the vessel, and then they release into the vessel. So that is not how we can overcome this. And we have seen in cell culture that we can improve this. Here is a double, here is a, here is this cup, which has brain endothelial cells on its lower bottom, so that it is a model of the blood-brain barrier. Now, when we put macrophages with amyloid beta into this side, this is divided. This is just a scheme, but we assume, so this would be loaded with amyloid beta. These macrophages cannot migrate across the blood-brain barrier. They get stuck here. Whereas if we degrade the amyloid beta, the monocytes can migrate across the blood-brain barrier and take the amyloid beta and the residual amyloid beta into lymphatics and get rid of it. Now, you will believe me when I show you the electron micrographs. So this is again the same blood-brain barrier model, but by electron microscopy. And you can see the brain endothelial cells lining the surface of the blood-brain barrier. And you can see a macrophage that's full of amyloid beta. We call it a fat macrophage. And this macrophage gets stuck here, does not migrate. But when we load, when we added monocytes that was not loaded with amyloid beta, it is migrating here through the pores. Okay, so how do we tackle the problem? We found that Alzheimer macrophages lack energy for their function to clear amyloid beta, and they do not degrade it. So we found that supplementation with polyunsaturated fatty acids like omega-3 fatty acids repair the deficit in the transcript of the energy enzyme and the function of macrophages in the uptake of amyloid beta and degradation. And the two enzymes which are upregulated are shown here: insulin degrading enzyme and metalloproteinase. Now, I will show you what happens when a patient is supplemented with omega fatty acids. We have on the next slide, we'll have a row in which patients' macrophages were supplemented, and in row S were non-supplemented patients. So let's look at this. So when we take monocyte, we actually separated single blood cells. We separated monocytes, neutrophils, regulatory T cells, memory CD4 T cells, and memory CD8 T cells. And we were looking for transcripts of mitochondrial energy enzymes. And so what you see, if this row under M are monocytes that are not supplemented, and because it's showing in purple means that there is all these energy enzymes which are shown here, which include glycolysis, citric acid cycle, oxidative phosphorylation, mitochondrial transport, and ATPase and nicotinamide metabolism. When these monocytes were not supplemented, they have very low expression of all these genes. But when they were supplemented, they are in red, which means they are highly upregulated in the enzymes of glycolysis, oxidative phosphorylation, and mitochondrial transport. And so actually, patient that we obtained the macrophages from a patient who had been supplemented with omega fatty acids. And again, here we see different oxidative phosphorylation and glycolysis and TCA cycle enzymes. And then we are looking expression of these transcripts in different patients. So these 6 patients were supplemented, and you can see that their transcripts are highly upregulated, whereas these patients and the controls were not supplemented. And you can see in blue that the level of their expression is very low of these enzymes. Okay, so how do we compare to approved therapies of Alzheimer's disease? So the classical treatment has been cholinesterase inhibitor, which prevents acetylcholine degradation, and that is, of course, still used. Memantine is another classical approach, but last year, the new arrival is aducanumab, which increases phagocytosis of amyloid beta by monocytes and microglia, just like what we showed in previous slides, but it improves this phagocytosis and transfer from plaques to vessels. However, it does not degrade amyloid beta, and the vessels develop cerebrovascular amyloid angiopathy. And the clinical results in post-approval, Aduhelm reduced cognitive decline, did not stop the decline. So obviously, the classical therapy are helping, but not curing the disease. Therefore, we are now trying to help patients by our experimental approach with polyunsaturated fatty acids. So as we said, PUFA upregulates the transcript of these OXPHOS and glycolysis energy genes and A-beta degradation enzymes. The PUFA increases the energy of these macrophages, and clinically, in patients who were supplemented, improve the cognitive state of Mencia patients from the score of 24 to score 25.8 in 1 year. Now, which supplements have been used and are being used? These supplements included a product from Norway and Sweden called Smartfish, which includes omega DHA, EPA, resveratrol, and antioxidants. And this drink has been superseded by a new drink, recovery drink, EO3, which in addition to these components also includes resveratrol, which of course is the French paradox in wine, that the wine is the cure in France, and thymoquinone, which is an antioxidant. And also commercial preparation, another commercial preparation is similar omega fatty acids, antioxidants, vitamin B complex, and nicotinamide riboside. So what kind of results can we expect? So we have patients in the study who had subjective cognitive improvement or mild cognitive improvement. And as you know, subjective cognitive improvement is just noticed by the patient. Mild cognitive improvement is when there are objective signs of confusion and disorientation and so on. But all these patients have activities of daily living. Now, when we looked at the MMSE on first visit and the last visit, we are seeing that SCR patients have gained, improved, whereas the MCR patients, some of them improved and some of them declined. And however, the progression to dementia did not occur in subjective cognitive impairment. In mild cognitive impairment, we have delayed the dementia up to 52 to 54 months, except this one at 26. So the treatment is helping to, in the first year, it helps to delay the dementia. And we hope that this treatment will be actually improved in the new drink, 3. So how do we know that this is working with immune cells? We have shown that the supplementation with omega compared to no supplementation changes many functions of macrophages. For example, again, the red amyloid beta is picked up when the patient is supplemented with omega-3. And the phenotype becomes anti-inflammatory, and the phagocytosis of this green fluorescent amyloid beta is much increased compared to the one without amyloid beta. Now, how do the patients fare over time? We have here the progression. That some patients improve, then stable, and then a slight decline, and then continue stable. These are actually years, to 2017 to 2020. Here's another patient, patient 14, that we have been very proud of. That patient, before starting supplementation, went actually almost to dementia. And as soon as she started to be supplemented, her mental state score improved dramatically and continued at close to even normal. But then she had a heart attack and she declined, and she died since then. Now, patient with dementia with Lewy body does not respond to this treatment, and we have seen continuing decline. So, in summary, we have evidence that macrophages of Alzheimer's patients do not work. They do not pick up inside the amyloid beta as the normal ones. And we show that the macrophages, instead of degrading the amyloid beta, release it into the vessels, which become congophilic. The red marks the congophilic nature of these vessels. And these vessels then don't transmit nutrients, and the brain suffers. And to put it all together, we know that the brain is being cleared by macrophages, which exit from the vessels, go to the plaques, load with amyloid beta, and then migrate back to the vessel. And then the crux of the treatment is whether we can degrade in macrophages instead of releasing into the vessels. And current work is now proceeding to find which enzymes will be increased by our new treatments, which will allow the macrophages to degrade amyloid beta, and then they can exit from the brain and with the residual amyloid beta. And we hope that that will lead us to the point which we are looking for, that there will be stabilization of this cognitive state. Thank you for your attention.
Yes, thank you, Dr. Milan. Is there any questions from the audience?
Sorry,
yes, sorry, go ahead.
I could not hear you well, but
okay, yeah, I have a question. I saw that somebody else want to talk. That in very interesting work, just wondering the dose of the purifier you used in a cell study compared with the dose or the amount of purifier that actually accumulated in the brain of the patients after you give them oral supplements. Did you compare the level of those purifier in the brain of the patients versus the amount of purifier used in the cell culture study? Are they the same level or they're quite different? That might explain the difference in effectiveness.
Okay, so our approach is to draw blood samples, isolate the macrophages, and test them in cell culture the way that I showed you. I will put that slide on again. These are patients' macrophages that were treated either with DMSO or the drink, omega-3 drink, and you can see that there is marked change in oxidative stress. The omega-3 treated have a low of stress. The mitochondrial membrane potential is increased after treatment with omega-3. The anti-inflammatory gene marker is increased after omega-3. The pro-inflammatory is decreased. And as a proof that this is working in the function, we show that these treated macrophages now are picking up fluorescent amyloid beta, whereas those that were not treated, if they were not treated, they did not pick up much amyloid beta.
Yes, I agree. Your cell study shows a pretty good effect of treatment with omega fatty acids. The question is the amount of omega acid you use in the cell study here compare the level of those fatty acids in the brain of patients after they taken the oral supplements. Are they comparable in the levels of those fatty acids?
Okay. Are these patients comparable? The treatment recommended is standard. There is a 200 ml carton of drink, which looks, I may have one to show you. It looks like this. The patient takes this drink 5 days a week, not on the weekend. Because if we have tested daily intake, and in that case, the receptors for amyloid beta become saturated, and it's working less over time. Whereas when we keep the standard dosing 5 days a week, one drink of this size, we have those results which we consider excellent. As I mentioned, so this patient, for example, is now like 5 years in the treatment, still doing well. Now, this patient, however, this patient number 14 had marvelous recovery. Somebody was very happy that she was able again to participate socially, and she went on until she had a heart attack. So, of course, this patient was actually 93 when she died. We treated her since age 89, and so we are very proud that we could improve her mini-mental score of 10 to a score of about 25, 26. And
yes, yeah, that's impressive. That's impressive. I agree. My question actually is, I think for example, this patient, they treat them with oral supplements. Did you take their blood, isolate their immune cells to see whether the ability, the immune cell from those patients treated with oral supplements, did their immune cells become capable of destroying A betas
okay.
or not?
So as you can see these dates here, we see patients in approximately 3 months intervals, 9, 12, 3, 6. On each visit, we are testing not only the cognitive state, but we are doing the biochemical analysis. We isolate PBMC, then we separate monocytes and we place them in cell culture. And in cell culture, the monocytes develop into macrophages. And then we can treat them in vitro as well. But we compare also patient over time, so that for here we do in vitro treatment. Let's see if we can enlarge this. So in vitro treatment with omega or DMSO, and you can see that the oxidative stress is reduced, the membrane potential in red is increased compared to this, the anti-inflammatory state is increased, and the phagocytosis is increased.
Got it. Got it. Thank you. Thank you very much. I appreciate it.
Thank you. So the biochemical results, if we want to repeat this, we can say that the biochemical results are complex because we are looking at all these genes in energy enzymes, but we have seen in different cell types, in monocytes in particular. So let's focus in monocytes, that if these come from a patient who was not supplemented or from a control, these expression of these is low compared to patient who is supplemented. And we also see that this is true for CD8 cells when these non-supplemented monocytes are in blue, meaning they have a low expression of all these enzymes. And when they are supplemented, as they are, many of them are, most of them are in red. And the larger size of the dot means it's more upregulated. And we can see that true for CD8 cells with most of the energy enzymes. So just the message from our study is that we are dealing with very old patients, some of them in their 90s. We have had patients 106 years old. And so there is both aging and Alzheimer's disease going on. But both of these processes finally relate to this problem, biochemical problem, that the energy enzymes are downregulated. As you can see in here in monocytes, the monocytes are in purple, meaning they are downregulated in all these energy enzymes. And this is true in other patients as well. And so we believe that the energy problem is both problem of aging and Alzheimer's disease, and that supplementation with omega fatty acids, which are transported by carnitine into mitochondria, which are the powerhouse of each cell, and they produce the energy around the clock. So we recommend that patients take their drink in the evening so that the monocytes are at their optimal performance during the night when the brain is clean, because the brain opens up at night when it doesn't have much to do. It opens up its spaces and it's perfused by cerebral spinal fluid. And this cerebral spinal fluid carries these monocytes, which are then optimally, they are improved by omega fatty acid drink, and overnight they work very nicely. And when we test those monocytes in the morning after the nightly dose, we find that they are still at their optimum level for phagocytosis of amyloid beta. So that is our recommendation. Take the drink at night, 5 days a week. And also we have a recommendation for sialic acid, because we found that sialic acid is a monosaccharide which is important for the receptors of macrophages for uptake of amyloid beta. And with this regimen, we see the results that I have already shown you, and that prolong these patients into very old age, mentally functioning. Thank you very much. Are there any other questions? If not, my presentation is finished.
Hello.
Doesn't seem that there are any more questions. Thank you for your attention.
Hello, doctor. Thank you. Dr. Milan. Stop sharing the screen, Dr. Milan. Is there any other questions? Yes. Thank you, everyone. With this, we are going to finish off day 2 presentations, Neuroforum 2023. Okay. Hope you will receive your certificates within 2 hours. And I request everyone to share your feedback as a testimonial. And we can meet up at Neuroforum 2024, which is scheduled in the month of March 27-28 in Valencia, Spain. Okay. Hope everyone had enjoyed the sessions. Please share us your feedback, which will update in the website of 2024, so that we can increase the scope of the conference in the upcoming years. Thank you so much. Dr. Milan, can you stop sharing your screen, please? Thank you. Bye-bye.